nice to meet all of you at this workshop. This workshop has emerged as some long-term discussions about simplicity and complexity in data analysis, in machine learning, in mathematical modeling, and so on. So in particular, between the organizers and speakers of this workshop. So intuitively, we can say that the data can be complex or the data can be simple, and the models of data can be also complex or simple. And in theory, we expect that these two complexities should match each other. But phenomenologically, sometimes, not always, but sometimes we observe kind of miracles. For example, sometimes it's possible to learn complex model from relatively limited data and even from few shots. I mean, it remains a kind of miracle. Sometimes increasing model complexity leads to emergence of some kind of self-simplification, sort of phenomenon similar to what is happening in statistical physics. And complex data in multidimensional space in high dimensions sometimes start to behave like very simple distributions. Sometimes, but not always, very simple principles lead to emergence of complex systems. So all these, I guess, familiar to those people who are kind of collected here. And the main point of this workshop is how we can approach explaining these miracles and how we can approach, make some simple steps towards maybe theory of this phenomenon. But in general, we will discuss questions related to complexity and simplicity in data analysis. So I would like to remind you that during the workshop, the microphones must be off. Unfortunately, I don't think we managed to set up a common chat. So the only way to ask questions would be to raise hands, and I will give a word to those people who ask questions. And this workshop will be recorded, as you have noticed. So at this point, I would like to give word to Ivan to introduce the first speaker. Please, Ivan.
Andrei, thank you very much. Well, I'm really privileged to make this introduction because it's really a great pleasure for me to introduce our first speaker, Professor Kees van Leeuwen, because I've been working on the case leadership for a significant portion of time. And I would like to say that Professor van Leeuwen started his research career and actually has traveled and worked across many universities, including the University of Nijmegen, the University of Groningen, and then the University of Amsterdam. And then he was appointed to a chair at the University of Sunderland, but actually that wasn't enough. And in a few years, Kees established a new laboratory at the other end of the globe in Japan, at Riken Brain Science Institute, which is a flagship Japanese research institute for advanced science. So, there've been significant progress made there, and I think this topic of today, this research program that we'll hear about, I think originated there. And for me personally, I'm really grateful to Kees for talking about that program. And coming from a somewhat different background, I was really enthused by visions for how to relate cognition, intelligence, adaptivity, dynamical systems, and perception in a single theory that I had a chance to think about and work on in Japan. So after working in Japan, Kees brought the laboratory back to European shores and established it in the University of Leuven, where he's got a research chair position. So, and now I would like to stop, and I'm leaving the floor, Kees, to you. We are very eager to hear about this research program and, of course, your talk.
Thank you for your kind words, Ivan. Can everyone hear me, and can everyone see the presentation? Yes, then I'm going to go to the presentation myself. Here we are. Yeah, as Ivan said, this is a long-standing project. It has been started at the Riken Brain Science Institute in around 2002, 2003, and it's a side project, but it is a side project that has survived the years and has continued to produce many new developments over the years. So I'm going to talk about this and take a little bit of a historical perspective. But first of all, I need to mention and thank all the people who have been working on this topic. The various people who were with me at the Riken Brain Science Institute, like Pulin Gong, who is actually the first to contribute to this project. Then number two, Daan van den Berg. And then subsequently Hoifei Kwok, Antonino Rifone, and last but not least, Michael Rubinov, who made an important contribution to the first stage of the project. Then at the KU Leuven, Nick Jarman, who worked together with Ivan in Leicester half-time on this project. Carlos Calvo Tapia and Manuel Hakiat, more recent additions, and most recently Elias Rensepiris and Jia Li. Jia Li will start as a doctoral student, hopefully in March of 2022 with us. Okay, to give you an overview, as I said, I want to take a historical perspective a little bit because of the long winding character of this project. So I'm giving some introduction. Complexity in neural microstructures is something that I will just quickly go over in order to present the basic idea of this project, which is adaptive rewiring. That's the catchword for this whole presentation. And the adaptive rewiring in our lab has a past, the first 10 years, so to speak, from 2001 to 2010. The present, which is in Leuven, 2011 to 2020, and then the future, which is about to happen and which has been showing some first contours, which I hopefully have the time to mention. So I have a lot to tell, so let me please continue very quickly. First of all, the brain, as we all know, is a very complex network. Its anatomy is not only very large scale, but also very intricate. And the large scale properties of this complexity can be shown in these connectivity diagrams, for instance. What you see is both clustering and you see a lot of off-cluster relationships. So basically, the brain, also in its functional connectivity, has a lot of structure. And this structure has some general properties that we know very well and have been very well established in the literature, both in the structure and in the functional connectivity. The brain complexity, the brain structure complexity consists of small worldness, modularity, and Rich-Klub's effects. We all know what this is. I don't need to tell you, this audience here, what we are talking about when we say small world, but I'm just going to present anyway the famous slides relating to this. Small world is the best of both worlds. You have local and global connectivity and therefore a very efficient structure in your network. Modularity, here we have it. The clusters in the network that are formed show communities that are connected through hubs, hub links, and hub nodes. Rich clubs, all these hubs are interconnected, not all, but tend to be interconnected in a way that they are very easy to communicate to each other. Yeah. Okay, so you have a distinction between rich club nodes and peripheral nodes, nodes that are not part of any rich club, but are probably part of some module. Okay, this is the structure, and this structure, of course, serves the efficiency of the brain's information processing, communication optimally. And the million-dollar question is, of course, where does this complex topology, small world modularity rich club, come from? Now, if this were the Human Brain Project, this would provide you with $1 billion answer, but we have only a low budget answer. And the answer is, this must be from adaptive self-organization. The brain cannot plan all this complexity in detail. It can perhaps plan the global contours of this complexity, and then this has to be tweaked or at least implemented through some self-organization mechanism that creates the precise structure. We believe that this adaptive self-organization is realized by the spontaneous activity of the brain. So the neural anatomy of the brain and the spontaneous activity sort of go hand in hand, although of course the initial growth of neuroanatomy precedes initially silence of the neurons. But once the neurons get to show some activity, they help shape the neuroanatomy, and the neuroanatomy is shaped in a way that optimizes the information processing or the spontaneous activity, if you like. So what does spontaneous activity look like? Well, here we have a nice sample. This is EEG recorded. You can see the top of the head here. This is in phase space. Now, what we see here is very beautiful structured activity. Yeah? Spontaneous activity is all but random. It is very well structured. What you can see is some kind of intermittency between traveling and standing waves of all kinds. Yeah? Some metastable character. The waves come and go and are alternated by brief periods of instability. Oh. In the initial stage of the project, we thought that this oscillatory activity was very important for forming these structured patterns, and that these structured patterns in oscillatory activity, the waves and the traveling activity, are actually the ones that shape the structure in an optimal way. So we started modeling this, and the very simple assumption was that we take the mass model of neural activity, a very elementary mass model in which this neural mass activity is governed by a chaotic attractor. We simplify that further by taking a section through this activity, and we simplify this even further by making it a logistic map. Now you see here that this is noisy and this is not, but if you couple logistic maps together, then they perturb each other's activity and then they are again noisy. So this is the activation function of a unit, but coupled they actually look more like this. So and here we have an activation function, a logistic activation function applying to each of these individual nodes, and they are coupled together, both locally and globally. Notice that this is already a small world. Yeah. Now, when we see what happens there, is we see a lot of activity again in phase space, and what you can see is that there at least some qualitative similarity between the brain activity and the activity in the coupled maps, and this is typical. This type of intermittent activity with standing and traveling waves alternating with brief periods of instability is true for a wide variety of systems. So this is not at all something that needs some fine-tuning of parameters, neither in the brain nor in such an artificial system. And this is of course very important because the evolutionary selection is likely to capture on things that are generic rather than on things that are highly specific and require fine-tuning. So this must be a very universal and robust phenomenon. Okay. Now we have the spontaneous activity. Now the question is, what is adaptive rewiring? Okay, we've seen a network structure where this spontaneous activity just runs amok. And here we see how this structure was designed. We have clearly a path here. The gray path is the one that we have in our design. But of course, that is not optimal for usage. There are shortcuts possible to get to the street on the right. And what you will see is that the use creates its own paths. Yeah? And as a result of that, the design is obliterated. The gray path sooner or later will fall out of use and will be grown with weeds and will ultimately disappear in favor of the user experience. Now we are doing something similar, which is this, very simple. We have a network structure, and we observe in the network where locally or regionally there is intense traffic between units. And where this traffic is intense, we create additional connections. Yeah, for instance, here we create a shortcut in the same way as we did in the part of the park in the last slide. So our idea was to use, and that was the first stage, yeah, which I call the past of this adaptive rewiring project. We used the oscillatory patterns in a network to drive the rewiring. Yeah? And for the oscillatory patterns, we used the coupled maps. The rewiring is adaptive. Every once in a while, we inspected the structure of the network. We looked at pairs of connected units, and we said which of these pairs are connected at that point in time, but not synchronized. Okay, if we find one, we cut the connection there, and we relay the connection to an unconnected pair where there is synchron. Yeah? So this is more or less how it worked. I can go in more detail if asked, but here this should be enough. And this is how it then works. Yeah, you see the activity and you see the spontaneous rewiring. Every 10th or 20th step, we stop the network briefly, look at the activity, and rewire. And what you see is the gradual development or growth of a structure. Okay, there you have it. You can see this is of course presented through a graphical trick that brings connected units together graphically. But what you can see is that there are clearly clusters, and that these clusters are interconnected, and that the interconnections between the clusters also tend to be interconnected. So yes, oops, this is what happens to a random network, and yes, these networks develop small worldness, modularity, and the rich club effect. Now here's some old publications. Not all of them are terribly old. The most recent one extends this project with weighted networks. So there are certain things we learned in this first stage. For instance, that the structure as it develops in this adaptive rewiring doesn't follow the instantaneous functional connectivity, not the instantaneous synchronies. It follows the, you could say, the slow functional connectivity. If you take a moving window over the functional connectivity, then you see something that seems to be driving the future structure. So we thought initially that the synchronization of oscillatory activity was very important, but it turns out not to be that important. It is more like the long-term slow processes in the functional connectivity, not the immediate oscillatory phases. Yeah? I have some stuff about network pruning, which I will skip for time reasons. There is an issue about spatial embedding, which is, I think, interesting, which I'll mention briefly. And then there are some issues about advantages of log normal weight distributions once you use a weighted network instead of a binary one. Okay. I already mentioned these things. I'll skip them. This is, I think, something to keep in mind. Yeah, of course, brains are not only optimized for their topology, for their connectivity, they are also optimized for other things, such as wiring length. You don't want to have connected units being physically far removed from each other. So, as in principle, you can see here in the topographical maps where everything that is close together in function is and structural connectivity is also close together in spatial in space. No? Now, if we do the adaptive rewiring and we impose a constraint on structure such that we define an embedding for the networks, that is, we place them all on the outskirts of a globe, for instance, then we can do something. We can, for instance, penalize wiring length. So a rewiring is less likely to occur, say, if the two to be rewired units are far apart in the embedding in space. Yeah? So when we do that, we can very clearly see how this develops from the very beginning. In the beginning, there is no relationship whatsoever between the networks between us, that is the hub status on the one hand, and the physical distance on the other. So on the y-axis, you have the betweenness. On the x-axis, you have the spatial distance. Now, on the first row, you can see that there is no connectivity, no correlation whatsoever. But with the exception of the leftmost, which is the column, which is the non-spatial one, where this correlation does not emerge, we see a clear correlation emerge between us on the one hand and spatial distance on the other. Yeah? At least in the linear and the exponential case, not so much in the logarithmic case. What does this mean? It simply means that the units that are hub connections are becoming long range in space, far away. Yeah? And the within module connections, the ones that have low betweenness, are spatially local. So we get local modules. Yeah? Here they are color-coded, two instances, linear and logarithmic, with the disforget about it. Linear is a case where the penalty for spatial distance is properly weighted. Logarithmic is one where it is underweighted. What you can see is that the modules indicated by a color code here on the left are relatively neat, so you get a kind of spatial homogeneity. Yeah? In the logarithmic case, this is far more messy. Yeah? You can see that there are lots and lots of ectopic units that are spatially within a certain domain, but functionally belong to some other group. Yeah? So messy in the logarithmic case, linear case, quite neat. Of course, not completely perfect, and shouldn't be, because there should also be some interplay between the modules. Yeah? So let's call this messy brain, well, there are different variants. Yeah? If we take the medium wiring cost as the norm, yeah, then we have a very simple model for what happens in the regular brain. Yeah, you have modules and you have rich intermodular connections. Yeah. If we underweigh the wiring cost, that is the bottom one, yeah, then we get this sloppiness. Yeah, we get a lot of intermodular connections, and this characterizes something in the brain that we could relate to processes of dyslexia on the one hand, but on the other hand, creativity. Yeah. There is an other extreme, which is where we overweigh the wiring cost. That is the top, where the modularity is extreme. The modules are very neat, but there are relatively few intermodular connections. Now, this is what I call the autism spectrum brain. Yeah? So as you can see, variants of this this evolution in adaptive rewiring are parametrized. Parametrized evolutions are possible. Brain variants are possible that are extremely interesting from the perspective of neuropsychology. Yeah? We could say that all these developmental disorders, such as autism spectrum, dyslexia, and so on, could be understood in principle as variants of brain evolution. Okay, so that was the past, now the present. In the present, we found, we took one important message home from the past, which is that the momentary oscillatory activity is not so important for the evolution of the system. So instead of modeling precisely the moment-to-moment activity, we could do some stochastic modeling in terms of diffusion. And that not only simplifies and therefore makes the model more elegant and more abstract, and we want our models as abstract as possible. Yeah? Details that are lacking cannot be broken and cannot be wrong. So we think that we should make things as easy, as simple as possible. Therefore, we try to change our approach towards activity to diffusion. So we know what diffusion is. But diffusion, of course, also takes place within brain networks. And that offers an elegant representation of network activity. Yeah? So what we do now is we have diffusion of activity through the connections of a network. So we do have diffusion basically on a graph. Yeah? And when we perform this diffusion, then we again get a parameter that we can use in scaling the network, which is this degree of centralization. Yeah? So what we do now is we let the network activity diffuse, but before it can reach a terminal equilibrium, we rewire. That is, we do a perturbation, and then after the rewiring, the diffusion starts over, and then again rewiring, and so on and so on. Yeah. If you do the rewiring in a very confined, if you do the diffusion in a very confined period of time, then you get what you have on the left as a product of the rewiring. You get a very extremely modular network. Yeah. If you wait too long before you do the rewiring and the diffusion has gone too far, then you get an extremely centralized network as a result. But then again, there is a critical zone within which there's room for a lot of variability, where if you just wait for the diffusion to evolve for the right moment of time, not too short and not too long, Goldilocks type, then you get this beautiful pattern you see in the middle, yeah, where you have modularity, but modularity at different levels of the hierarchy, and you have a lot of intermodular connections as well. So this is a nice hierarchical structure that we typically find in brains. And yes, again, it has all the properties, small worldness, modularity, and rich club. Another thing is in this diffusion scenario, you get a slightly different perspective on spatial embedding, because the diffusion can now also take place not only in the network, but also in space. And then distance starts to matter, and also the field of the diffusion starts to matter. Yeah? And we can combine, once again, the adaptive rewiring according to the network diffusion with rewiring according to distance, or rewiring according to the alignment of the direction of diffusion. So these three possibilities are there, and you can, of course, do one of each, or you can do a mixture of both. So first one is exclusively distance. Yeah, you see that as the second column from the left. And there you see that modules tend to be formed, but there is little global connectivity. The network typically is not a small world. Yeah? If we're talking about field, then you see that the alignment is nicely in a particular direction in space. And then if you do diffusion, you get the usual thing that the networks are nice small worlds, and of course have all the other trimmings, but you cannot see in the morphology any structure. Yeah, you need to go back to the case where you have the functional view, where you can see the clusters. So you don't see, I mean, the morphological view, just how things are projected in space, doesn't change very much. It just gets, it just looks chaotic. However, if you take the view where you have the connectivity diagram, you will only then will you see that the structure is optimized connectivity. Yeah. So here we have the idea, yeah, mix diffusion-based rewiring with those distance and field principles. Yeah. And if you do that, you get some nice. I should stop here. You get some nice structures that really resemble some of the stalwarts of the neuroanatomy. Yeah, you have here, for instance, you have distance. Yeah, this is a system that is minimized for distance, and at the same time a small world and a superchain. Yeah. Here you have spatial alignment, and alignment can be sort of parallel, or it can be going in out in concentric waves, and you get very different morphologies based on this. However, their structural connectivity in terms of network is still small world, modular, and rich club. These things are from the present, and here what we have seen in the last few demonstrations is that we can actually try and begin with this very abstract model to mimic certain developments in the brain, in the biological brain. Yeah, at least the structures that we can generate start to look like anatomical structures. But we are missing still a very important ingredient, and that is there in the future, yeah, which is when we use diffusion, like we used oscillations in the past, our networks are typically undirected networks. Yeah, they can be weighted, but they are undirected because synchronization means A is synchronized to B in the same way as B is synchronized to A. Yeah, and of course the diffusion depends on concentration, but the diffusion is equally strong from A to B as it is from B to A, and therefore all these networks are symmetrical. Yeah, so brains are typically directed networks, And in order to describe the evolution of the microstructures and motifs, for instance, and apply these adaptive rewiring to biological, but also to artificial neural networks, we still need one ingredient, and that is the directedness of the connectivity. Okay, and we found a solution, and that is Elias Renseker's work in applying a directed generalization of diffusion. Yeah? So once again, we are using diffusion, but we are evaluating the diffusion only depending on the incoming or the outgoing activity. This is respectively consensus or advection. Yeah? So both consensus and advection are generalizations of diffusion, but regarding the activity only, evaluating the activity only in one direction, only looking at what comes in or what goes up. Now here we have what we have in those cases. Yeah, when you use advection, as you can see, yeah, we only rewire the out degree neighborhood. Yeah, and then of course, as a result, what forms are out hubs, that is, hubs from which activity diverges. That's the top row. When we use consensus, we rewire the in degree neighborhood, and therefore we get input hubs, that is, convergent hubs, hubs from which activity out of the neighborhood converges. So, of course, networks where everything diverges or where everything converges are not very good and not very useful. So what we do is we take a balance between advection and consensus. And when we do that, we see that we get a very nice situation where the system rewires to produce both input and output hub, both convergent and divergent hubs. Now, why is this important? Well, this is important for a number of reasons. Still the network has modality, still the modularity, still the network has rich clubs, but now the network also has something principally new, something that plays an important role in neuroscience, and that is the enigma of how to do organized context-sensitive processing. Well, the solution proposed in theory by Kumar and Aartsen, Shaw and Scheibel and others is the so-called convergent-divergent units. What are convergent-divergent units? Well, this is the idea. You have a convergent hub at the beginning and a divergent hub at the end, and in between you have a couple of intermediate nodes. The convergent hubs collect the information in a context-sensitive manner. The intermediate nodes act as a processing unit, and the divergent hubs basically broadcast the output of the computation in the intermediate nodes to a broad set of targets. Now, the interesting thing is that our adaptive rewiring, using this mixture of advection and consensus, automatically and spontaneously generates these convergent-divergent units. And not only that, they can do this in a way that is, again, has a nice tunable parameter, namely the degree to which the intermediate nodes themselves are isolated from the rest of the network. So those convergent-divergent units exist under balanced convection and consensus and advection. The members of the units, that is the intermediate nodes, are more densely connected than peripheral nodes, nodes that are not part of a unit. Therefore, we get fully connected motifs in the convergent divergent units. We see that there's still the rich club and the modularity features. This is what constitutes the convergent divergent units in our system, and those convergent divergent units we can claim could provide an important functional role for a system like the brain. They could be considered as the computational core of it. And also, to make things even more elusive, there is the concept of global workspace in consciousness. If you take the whole of these convergent divergent units, or a proper subset of them, the ones that are, let's say, the most globally connected, then you have something like an architecture for the global workspace. So the lessons for the future is that the emergence of convergent structures are possible in this work, and that the embedding of these hubs create motive structures. These are some publications on the way. The first one is just accepted. Thank you, Ivan. And the second one is hopefully going to come soon, but is already available on arXiv. Conclusion, adaptive rewiring offers a versatile principle to elucidate how brain connectivity structures could emerge. Of course, this is a just so story, but I think a just so story is sometimes important. You need a simple story in order to capture at least the beginning of how this brain how this brain development works. And of course, when we learn more about the evolution and the development, this could of course be interpreted in terms of a more realistic model just to come. We have a number of testable consequences for this model, such as the complexity, of course, the presence of varying brains, and the presence of convergent divergent units, as well as the theory that these are the foundation for the global workspace, which in terms forms the important functional architecture for conscious experience. Thank you very much. That was it.
Thank you very much, Kees. It was a super interesting talk. We have two questions. The first one from Alexander Gorbay, please.
Hello. Hi. Okay. So then I have one problem. When I'm discussing any type of neural networks,
yes,
natural, artificial, for me, before all discussions of structure, I should answer two questions: the structure of data world, which works, and the structure of the task world. Because without that, I cannot understand why and what for should be some structures in the brain and the neural networks before I specify this structure. Moreover, this is the first part of my question. Could you please express your opinion about that? And then I will with the second part of the same question.
Well, I said before that I could, of course, pretend not to hear your question, but I think this is a very important question and a very important issue. Of course, with artificial neural networks, these are customized and optimized for specific information structures and for specific, let's say, functional purposes. Well, the brain is not. The brain, I think, is a general purpose system, and of course, it is therefore to be optimized for information processes, but not further to be specified at this point or at this moment. That is one. So all the further specification is secondary to the setup of the global characteristics of the brain as something that's optimized for some communication purposes, which are not yet to be developed any further. This is why we believe that the brain is developing not according to specific information processing tasks, but according to some general communication that takes place in the brain. And this general communication takes place in spontaneous activity. And we do see this spontaneous activity in various times, at various times during brain development.
Okay, so excellent. Therefore, exactly after that, the second part of my question. Of course, I can completely agree that brain is not specialized on very narrow set of tasks. Absolutely. But I should say that the world of our data has very special structure, in the sense that this is not from the smooth random fields or some random variables. This is sort of hierarchical that there are things. It's many important words. There is ontology, and things are organized hierarchically. Things consist of things, and there are some attributes and some relations. And this hierarchical replacement world is very different from anything that the standard probability picture with reliability gives us. Therefore, I should say that in your ideas of structure, I can see some hints to that structure of the world. But for me, this specification of general, not of the specific, of general properties of our world and general request, because our brain is not constructed for working in smooth fields. It's constructed to work in this world of hierarchically organized things, but some general properties should be somehow assumed. This is my opinion, but please, your comment to this opinion.
Well, I mean, the short answer is I fully agree. Yeah, what I'm describing is how the system evolves under random fields, if you like. And this is of course only part of the story. At best one half. Yeah. Why have I started doing so? I have started doing this because I felt that the approach that only looks at learning of particular solutions to particular problems or learning particular associations in data in the typical way it is done in the psychological literature, where we call this connectionism. Yeah, this is very unsatisfactory to me. Yeah, I mean, the spontaneous activity is there from the very beginning, and it must have a role. So I reasoned. So this is why I developed it in the way as I did, as a kind of counterpunch towards the anything that is specific in the data structures that we actually learn. Now, this being said, of course, my approach is just as one-sided as the approach that exclusively looks at data. What I should do, therefore, is I should give you a proper handshake and says, yes, of course, we need to look at these data structures and see how these data structures are influencing the generation and the shape of the network. Yeah, this is of course something that we want to do and that we hope to do and have already started doing in a very limited way, because one of the papers that is just out or just being in press in network neuroscience is trying to show how the system that develops under spontaneous activity is modified in its structure by incoming information. Now, we have only done the very, very first preliminaries of that and published that, and to show that it actually still works, that incoming information doesn't completely destroy, for instance, the evolution of such networks. That is already something for us, yeah, because combining this spontaneous activity with information is in fact for the system a random perturbation at this moment. So we should look at this more carefully and more in detail, and hopefully we have time to discuss this in the future.
Thank you.
Okay, and
Let us discuss.
thank you. And a quick question, if possible, from Ivan.
Yes, of course. Well, actually, I had 3 questions. 2 of them have already been asked and answered. So I therefore would like to go with the small and technical question case. It's fascinating what you actually found, that there are those incoming and outgoing hubs. And like from our perspective, if I may, the importance of them is very high, because like they could be those sort of grandmother cells, or like those cells on which the information is converging. So like and they indeed can be acting as abstractions or switches if needed. And therefore the question is, have you observed in practice, or like in theory, a possibility for you to somehow control the number of those, or maybe there could be some relationships between the scale of the network, the size, and the number of these hubs? Like, is there any sort of intuition or like hard facts around this?
Yeah, we have a lot of. I mean, let's start from the other end. What we try to do is to make the networks as simple as possible to do what we want them to do, because we don't want to have all kind of randomly engineered stuff in our models. We just want to see how stuff emerge, how things emerge, and then have the minimal tweaking parameters or control parameters just come up as we develop the model. Therefore, we are often surprised by the versatility of this approach. We sometimes just discover what the control parameters are. And yes, indeed, we have by now quite a number of ways to tweak the model, as I have tried to specify during my talk. Yeah, and in this case, for instance, yeah, you could say that, for instance, the distribution of hubs, both input and output hub, converging and divergent hubs, is one parameter that we can easily control. First of all, the proportion of input and output hubs is just how often you do consensus or advection, respectively, and that is one important parameter. Another important parameter is how often do you do this rewiring randomly. Then we also always had a certain proportion of random rewiring in the network, which is important. And this random rewiring could be done quite infrequently or quite frequently, but you need to do it from time to time in order to secure the global coherence of the network. Now, that parameter is one that we can use to tweak the total number of hubs, because as intuitive, if you do a lot of random rewirings, you preserve much randomness in your network, and therefore you simply have less hubs. Yeah. So that answers your specific question. I should say one more thing. We are now using spatial embedding for these models, and spatial embedding, of course, complicates things a little bit more. In the spatial embedding, we have no randomness, we have no need for randomness. Instead, we have just the spatial embedding. The spatial embedding is random with respect to the structure, and therefore we can use the spatial embedding to similarly tweak the network.
Thank you.
Okay.
My pleasure.
Thank you very much again, guys. I think we have a possibility to upload the in Teams. I don't know if you see. And let's switch to the second talk of this first part of the workshop. It will be given by Ivan Tyukin, who is a professor of applied mathematics at the University of Leicester, at School of Computing and Mathematical Sciences, and he is a fellow of United Kingdom Research of Innovation Turing AI program. So at the University of Leicester, Ivan is a deputy director of the Center for Artificial Intelligence, Data Analytics and Modeling, and he leads the Digital Intelligence Systems Facility as a part of Meteor researching program at Space Park Leicester. And also he acts as an editor of several journals, including Communications in Nonlinear Science and Numerical Simulations. His research interests are in, he has very wide research interests, including mathematical modeling, dynamical systems, adaptive control, artificial intelligence, data analytics, machine intelligence, and learning. So I'm very happy to give a word to you, Ivan. If you can fit into kind of 35 minutes, would be great. But please.
Thank you. Thank you very much, Andrei. I am timing myself now, like there is a clock next to me. So it's a great pleasure to talk, and I would like to say that this is the presentation is a joint work with Alexander Garban, who is present in the audience, and who is the real director of Artificial Intelligence Data Analytics and Modeling Center. So we are also building on some other works in the past, and some names I will be introducing as I speak. So the topic is quite a, and the title is quite a provocative, the mathematics of learning from high-dimensional low-sample data. And I would like to start the talk with some clarifications, because I think we need to understand what does it mean, low-sample and actually high-dimensional, and why is it at least important to take a look at this. So I'll start with these slides where these pictures, both on the left and on the right, are taken from the paper which we jointly written, and the reference is provided at the bottom of the slide. And but actually, I would like to start with the first reference about Donohoe's lecture made 21 years ago on the high-dimensional data analysis. So in that lecture, a picture was presented whereby we have a classical area where, let's say, central limit theorems work, where which enjoy the benefits of concentration inequalities, or actually even infinitely large, or like at least presumably infinitely large number of samples relative to the number of attributes that have been measured. And there is this post-classical area, and the post-classical area, according to Donohoe, is where actually we don't have this luxury anymore. Importantly, though, he specifically points to the case where the problems start to get a little bit even more difficult when we have the cases when the number of samples is significantly smaller, or just smaller than the number of than the number of attributes that we have. And there are many practical problems in this case. Like, for example, if people here are present in the audience who work with medical data, they will immediately see that that's indeed the case, because even if we look at a standard medical record of a patient in NHS, a National Health System, then these records can reach, in terms of the number of attributes, to thousands of attributes. Although when people do research on this data, normally you don't have the luxury of having infinitely many. What you have is that you have hundreds of records at your disposal, and maybe thousands. And it's not because this data is not available, it's just because it's very difficult to clean and properly represent and make this data accessible and available. Now, the picture on the right is what our vision of this post-classical area is. And in fact, we stay away from the asymptotics here. And again, we replace in this vision the the number of attributes with dimension of the data set. And according to this picture on the right, the classical area is where the number of samples is basically exponential in the data dimension, or exceeds that. and like, please don't pay attention to those constants A, B. I presented them just to indicate that there are those constants, but unfortunately you cannot play with these constants a lot. So they're fixed. Sometimes they're just 1/2, sometimes they're like a different number, but in fact they're fixed, and like there is nothing you can do about them. And there is this post-classical area, which is in fact kicking much earlier than what de Noga envisioned, and it kicks just beyond this exponential bound. And there is this empty area, and the empty area in this vision is the case where like basically we don't meet these situations in practice, because even if you take this dimension of the data, let's take this simplest definition as the maximal number of linearly independent vectors, then of course dimension in this aspect cannot be larger than the number of data samples that you have, right? So so let's move on. And my next point is that the classical domain. So like, you know, this machine learning and AI and data science as we know to date, all the theory been underpinned by mathematical machinery that belongs to the classical domain. So we are using central limit theorems, we are using law of large numbers, we are using concentration inequalities if we have uncertainties about the data distribution, etc. And these are the machineries that actually are extremely useful in this area, and this is upon which a large body of modern machine learning and AI is built. So unfortunately, as I said, when we go to post-classical domain, and why is this important at all to pay attention to these cases, is because sometimes when we escape and break through these exponential boundaries, you know, things may go wrong. And like as an example of like where things may go wrong, or like not wrong, but actually sort of out of our traditional naive expectation, is this important Martian capacitor law, whereby we, let's say, consider an example like where we have n capital measurements and d attributes, and we have those measurements and suppose that the entries of those matrices are like have all zero means and variance equal to sigma squared. And maybe even for simplicity, we can think of the sigma squared being just 1. And then we do, let's suppose, do this principal component analysis, and like we compute this matrix Yn and calculate the eigenvalues. Now, if we let n capital go to infinity, then of course our expectation is that things are going to concentrate around like sigma squared. But if along with that we let, let's say, d go to infinity as well, so basically making the dimension large consistently with the growth of the number of samples, then we have this Martian capacitor law, which says that in fact the spectrum is not going to be concentrated anymore, and like there will be a pretty spread out distribution of the eigenvalues. And like again, like to put this in perspective, like these are examples of what's going to happen when we fix the ratios and start to vary it from, say, a reasonably large value of like 1.62 to small. And you see that the support of the area where the of the measure where the whole thing concentrates starts to shrink when lambda goes to 0. But that's exactly the case when lambda goes to 0 is a case where dimension is fixed and the number of experiments go to infinity, right? So like, but if we break through that, there is no longer agreement with our naive intuition that we can somehow expect concentrations. So, and why is this important? This is important because many instances of practical data sets, they actually are already in post-classical domain. And in fact, even if we talk about like a previous definition of post-classical domain, which I gave earlier, like even conventional data sets around, like say, face recognition, where people believe that the data is in abundance, in fact, it's not, because we are already working in the post-classical area. So, but again, like markedly and like more importantly, in many practically important data sets, for example, medical records, we are near the boundary or on the boundary of the empty area. And here is an interesting question, because I'm trying to plot a monster story, but what happens in reality is sometimes not necessarily the monster. So like there is empirical evidence that learning close to empty areas as well as on the boundary is possible. And like, for example, we are seeing these papers about few-shot learning, one-shot learning, etc. And like people are successful building these systems. So therefore the question is, why is this possible? And when things can be possible, and what mathematics can be used to bring some clarity in this space. And therefore, I will focus primarily in the rest of the talk on two subclasses of the challenge, which is basically learning from few examples in high dimension and learning from scarcely labeled data. So they are related, but they are not, of course, identical, those questions. And I would like to start with the first question, which is learning from a few examples. And I would like to consider a classical setup, which is nicely presented in those two references, which I present on the slide. So suppose we have a feature generator. Feature generator can be any existing, let's say, legacy model. It can be a deep learning model. It can be anything else. But the important thing is this. So it takes the data and it generates features, right? So suppose that we have that. Yes. And then the thing is this, like we have few examples of the input data, and let's say they belong to, say, class 1 or to dogs. And then we put this through a feature generator, and as an output, we generate, let's say, a prototype, or in this particular case, an empirical mean of those features. So we can take another set of examples, and again, like I deliberately illustrate here with 3 examples. Cats, we put them through the feature generators, generate features, calculate empirical mean of those features, and then we do the following thing. So we generate an example, and that example goes through a feature generator. And we do the following thing with it. So like we compute inner products of the features generated by this example with the empirical mean generated by our previous experiences. And we do the same for the second class. And then we decide, let's say it's class 2, if this inner product between the feature and the empirical mean is bigger than the inner product between the feature and empirical mean of the first class. Okay, so this is a very simple thing, and this is a very simple scheme. But apparently, as these papers report empirically, it works. So like we can actually do construct those models, and we can learn from these representations. I will later comment on and define properly what does learning mean in this context, but for now, that's I would like to stop and to not explore that. Moreover, there is a very interesting, again, like a result here. The accuracy is quite remarkable, as you can see. So like 5-shot learning gives you accuracy about 70%. And that's really astonishing. So like that's what I think is really remarkable, in fact. So the question is therefore, why does it work at all? Like why this works? So there is a related question, and I think Andrei kindly mentioned this in the introduction earlier. Look, we have these big deep learning models, and we use, according to what we have, according to like earlier definitions, we use the number of examples, which is incomparably small to the complexity of the model. If I say associate complexity with the number of parameters, right? So, and sometimes these models actually generalize well. Now, importantly, when we feed the same models with the random data, right? Basically providing noisy labels, requesting that this noise is reproduced. Of course, the generalization doesn't happen, and in some sense, it also very nicely correlates with the early discussion that we have that apparently there could be some structure in the data that helps the thing to survive, and maybe that structure does not require these complex models. Or maybe these complex models do this structuring of the data in a certain way. So therefore, the question is, why do these models generalize so well, and why they don't at times? So actually, there is a more recent version of that same paper published or like republished recently, where the authors made some other experiments and they tried to understand like what was going on. But I think I would like to carry on further. So now let us speak a little bit more formally and try to bring some mathematical understanding into this set of questions. And again, like the question is, how can we learn from high-dimensional data? And now I'm going to define what does it mean by learning when I say learn. So we consider a generic setup where we have an input, we have a latent space, which is important for these schemes, and we have a label, right? So like these are all maps. F is a map and G is a map. So and we have a data sample that's drawn from some distribution. So that distribution, we suppose, exists. Which is already a strong assumption. Later on, I will be assuming even asking even more from the distribution, but for now I think it's okay. So and I would like to consider two problems. Problem one, which is the problem of learning few examples, and problem two is learning from few examples. What is the difference? And the difference is as follows. So for the first problem of learning few examples, we consider a classifier, we consider a new set of inputs and a label that is going to be associated with this new set. And what we do is that we pick this number PE to be a number that quantifies the quality of learning. It's going to be a probability, in fact, that has to be guaranteed in some sense. And then what we want to do, we want to find an algorithm that produces a modified function from the latent space into the space of labels, so that the classification on the new set of inputs is as we request, right? So it has to be exactly this label. However, on all other data which are randomly independently drawn from this probability distribution PU, we should have that the classifier is not really far away from where it was before, right? So we had this sort of benchmark behavior, which is determined by the map F, right? And what we want to do is that we want to minimally disrupt this benchmark behavior by introducing this learning thing. So, and this PE, if it's equal to 1, then of course we will be with probability 1, not disturbing anything, right? So now problem number 2 is fundamentally different. So in this fundamentally different problem, you see like the setup looks pretty similar from the outset. But what we request is that these are drawn now from some probability distribution. So like these new examples are drawn from some probability distribution. And we have a new label that's associated with this set. So instead of a single number PE, we introduce the second number PN. And what we request is this. So we request that the classifier now performs as we want it to perform, but not only on the training set, but also on all possible samples from the new set with certain performance indicator. And that performance indicator is quantified by the number PN here. And similarly to the previous case, we actually request that by modification, by modifying the classifier, we are not damaging the property of the system that's been given to us before, right? So like the difference between the two settings is this: in the first case, we absolutely have to provide prescribed response to the new set of input data. And in the second case, we should generalize beyond the training sample, okay? So, and these are two different cases. Now, what about assumptions about the data? So I would like to give two assumptions, and these assumptions are not, of course, limiting the theory in any way, because they can apply but let me go through that. So the first assumption is this. So the probability density Px, which is induced by this distribution of input, because remember that X is a latent space, right? So like the density exists, is defined in the unit ball, and there are constants Cx and R such that the density is bounded from above by this number. So what this condition asks for, essentially it asks for, like, we don't need the density, in fact, but what it asks for the absence of pathological concentrations in the data. And that has connections, of course, with the data dimensionality explicitly now. So, and the second assumption is that basically the probability density, which is induced by the map F, for the new class also exists, is defined in some ball centered at a point C and whose radius is V, right? And again, there is a non-concentration requirement here that it doesn't pathologically concentrate, right? So these are the two conditions. And of course, as I said, like previously, we can generalize the case to not necessarily these balls, but we can work with cubes, we can work with ellipsoids, etc. But the essence of the requirements will be the same. We do not want that the data is concentrated, and that there is some requirement that prevents that. So and now I think I'm ready to present the first result. It's theorem 1, and it's presented in our latest paper, or like archive version is presented here, but it's in IGCNN 2. And it's also a joint work with Mohamed Al-Hudaydi and Chinhua Zhou. I think Mohamed is in the audience, in fact. Consider a classifier F and let this U be a finite set, and consider like that we have a label. Now what we do is this: we calculate an empirical mean of the latent representation. And what we do next is we construct the classifier now in the following way. So we assign the label L nu to an X if the following inner product is bigger than some threshold value, and of course we have to pick the threshold within some range. And of course I have to say that this number needs to be at least positive, right? Then what comes out pretty straightforwardly is that this is a solution, in fact, to the problem of learning few examples. And PE is determined by this number here. So the good thing about this number, and like, is that when the dimensionality of the latent space in terms of n is large, then this term vanishes exponentially quick with dimension. And that means that like the higher the dimensionality, the better the capability of the model to learn under these assumptions, to learn few examples. Now, and again, like the argument is geometrically very simple, and I'm presenting it with this diagram here. So suppose that we have our sort of space, which is determined by this ball with radius r, and we have these data points, which are red points here. and blue point is the centroid. So like what we want to do, we are essentially we want to calculate what is the volume of this spherical cup, and as you can see from these calculations, it follows like a very simple laws and rules, and there we go. We have this exponential dependence as we want. So there is something else which I want to add to this one. Because essentially not only these data points which have been learned in this regime, but also all the convex combinations of those data points, of course, are going to be learned. So if that's the intention, then this is going to be guaranteed by this scheme. If this is not the intention, then we'll have to do something about that, right? So, but again, like you can argue, like as I said before, this is just a memory, and there is nothing about learning. So, like, can we do problem 2, please? And the answer is yes, we can. And for this problem 2, and before I go into that problem 2, I would like to say a few words about some other properties which are going to be essential and important for what's going to happen. So the first property which I want to state is the property that of near orthogonality of vectors drawn in n-dimensional domains, and in this particular case, n-dimensional unit sphere and equidistribution. So the property is this, and the precise formulation is given in our paper in 2016 in information sciences, and like again, like a modified version that suits the purposes of this talk is presented in this second paper there. So what this result states is this: if we have this equidistribution and start independently drawing from that equidistribution, then we will be getting exponentially long corteges of nearly orthogonal high-dimensional vectors. And the interesting thing is that this exponentially long corteges will be such that the number of things which are or vectors which are pairwise near orthogonal will of course exceed the basis of the space. Right? So the result in this respect generalizes earlier works in this area by Vera Kurkova, who published this paper on the existence of exponentially long quasi-orthogonal corteges in 1993. What we are saying here is that not only they exist, but they are also typical, right? And then here is just to signify this statement. Here is my simulations. Here are my simulations, which are made with hypercube, of course, and n is the dimensionality here, and n is the number of samples. And what I'm plotting here, I'm plotting, distributions of this quasi or nearly orthogonal corteges, and you see that when the dimensionality is sufficiently high, I'm actually exceeding the number of, I'm exceeding the basis, right, with high probability. And that's just to tell you that that's happening. So, but the important thing is this: the phenomenon of near orthogonality plays a crucial role in this next statement, which I would like to call the law of high dimension. And we've been discussing this in our group, what is the best name for that. But in essence, it's the following property. So suppose that we have a set of ID random vectors drawn from a distribution satisfying assumption 2. Remember, the assumption is that we actually say that no concentrations occur. And let's pick two numbers, delta and epsilon. Let's pick them arbitrarily in an open interval from 0 to 1. Then what we do this, we calculate the empirical mean of the data sample. And then the following happens. The probability that the empirical mean is within close to the center point C, right? And the closeness is defined by this upper and lower bound number here, is this. So the probability exceeds this number. And what happens is that if the dimensionality is large enough, then we can always pick delta and epsilon small enough so that this probability is still large, but then the empirical mean and the center of the distribution where this thing is sampled from are sort of in the relation of the following type, that you are going to be about like v squared, or v over square root of k away from the point C. And remember that this point C is quite arbitrary, right? So like it's not an expectation, and that's what I'm going to say. So like and the most important thing is this. Given that if I can let the n go to infinity, or like make it sufficiently large, then the bound becomes pretty tight, because you see from the upper side it will be bounded by this one, but also from below it will be bounded by this number, right? So and then here is the slide with interpretations of this result. So in contrast to the law of large numbers, as we know, this law does not guarantee that the sample is close to its expected value of the random variable. Instead, we are saying that the sample mean is to be close to some arbitrary point C. That's a very interesting thing. Of course, it's not an arbitrary point, but that C is not, of course, an expectation. But that C defines the distribution in some sense. Now, and again, like remarkably, if you let the n grow, then the bound is pretty tight, right? So like you cannot be much more off this v over square root of k, but you cannot be actually much closer to C than this number, okay? So, and here is the statement which builds on this low high dimension. And the statement is this, I would like to skip the complexity of the preamble and focus just on what the theorem says. So suppose that we satisfy all assumptions, then we take the empirical mean as instructed, and we take exactly the same logic of constructing the addition to our legacy system. Then this map G star is a solution to problem 2, and now remember, solution to problem 2 is much more difficult apparently on the paper because we have to now learn, not necessarily memorize. It's a solution to problem 2 with these probabilities. But now again, what I highlighted in red here, if we pay attention at the dimension here, if the number n grows, then these values PE and PN can be made in some sense arbitrarily close to 1. So we can be successful if the dimensionality is large enough and the data is good enough. So how this argument works, and again, the argument works along the similar geometrical principles as I described before, but now there is this place where the law of high dimension kicks in, because if we knew the exact positions of the center, then we could just draw a line between the two and separate the two. And in high dimensions, we would be able to succeed, and the conditions of when we are succeeding are presented in one of our, I think it's in that paper. The problem here is that we don't know where the center is, and this is where the sort of law of high dimensional is being used, because yes, we may not know where the center is, but what we are guaranteed to know is that the empirical center is within some prescribed distance from that center, and we are going to use this information to produce the probabilities and to produce this performance metrics. So that's how the machinery works. So there is something else which I wanted to say, but I don't think that I've got time. And let me now skip this for the next thing, because what I wanted to talk is a little bit about cases where we work with scarcely labeled data. I would like to go to experiments now. So nice theory, but does it work? So the answer is yes, it does work quite, because this is what the experiments which we've done in our group. So we took a standard CFR10 data set and trained it on 7 classes. Yeah. And what we wanted to do next is to let's check how this training can be used to learn classes 7 and 9. And voila, that's what we are getting. Like the system didn't see any of the examples from class 7, 9. And then we've got 10 examples. And after these 10 examples, we were able to, in fact, learn these new classes. And here are RSE curves for that. And similarly, we took this VGG16, we looked at we trained it on 70 classes. And this training was done on the 70 classes. But then what we've done is we look at classes 90, 99, and used latent spaces in this VGG to basically repeat this 10-shot learning. And as you see, the result was quite successful. So now I think I'm skipping this one, and I'm going directly to the conclusion. So the conclusions are the following. So high-dimensional data indeed presents a significant challenge for modern machinery of machine learning, AI, and data science. And it's because in reality, we are forced to be in post-classical domain where conventional theories, as we know, may not always work. So in addition, however, to statistical laws that underpin the body of ML, machine learning and AI that we know, there are laws of high dimension that can be used and which are practical. So these laws, in some sense, and I think I tried to convince you, they are already capable to explain why this few-shot learning may work. And they also explain when generalization is possible in over-parameterized models. So the important thing is that dimensionality of the learned or like defined otherwise feature vectors is the key for fast and successful learning, and subject to, of course, data distribution satisfying the same assumptions. So, and what I would like to state in the end, I hope that this gives some inspiration and paves the way for potentially a unified theoretical framework, if it exists at all, to address the problem of learning in high dimension. And that framework can be at the intersection of the concentration of measure theory and geometry. And I will finish at this point with this list of papers. If you want to take a look, I will make the presentation available afterwards. Thank you very much.
Thank you, Ivan. So do we have questions in the audience? So when people think, maybe I can ask a simple question.
I'm already afraid simple questions are difficult to answer.
No, I mean, it's really kind of, in my kind of naive point of view, the simplest example of when few-shot learning is possible is when you have very well-defined clusters in the data, and then you just need a couple of samples from each class to. So, I mean, this kind of high-dimensional view on this picture, does it bring anything more to this, or it's how to say? So
well,
are there any? Yeah.
Yes, I think I understood, Andrei. I think I understood, because I think that's the key to. Let me go back to this business. I think the answer to your question is in assumption 2. Mm-hmm. So that's what it is. In some sense, it's a yes and no, because indeed the best learning occurs from when? From just like if the whole data is concentrated in a single point, right? Like, but that's somehow a violation of assumption 2 now.
Yes.
Right. So on the other hand, what this assumption 2 states is that if there is a lump of something, and that lump has the property that it can be placed into a ball, and you can then have this additional property that if you place that lump in a ball and define a probability distribution on that, that will not be pathologically concentrated, then you can learn from that. So I think that's the bridge between the clustering and that assumption. So yes, in that respect, it's right. So if we define cluster as having this sort of lump in the data, then yes.
Mm-hmm.
It just somehow cements in the formal statement that that's how it can work.
I see. Any other questions from the audience, please? Don't hesitate. Raise your hand. If not, maybe we can make a short break here. And let's come back at 3:45 sharp, please.
So, Andrei, and what about coffee?
Yes.
Do you provide beverage coffee, some snacks?
I provide unlimited virtual coffee to everybody.
Okay.
Oh, I was looking for a knock to my door. Like I was looking for a delivery.
Okay. So, assumption. Okay. I cannot, Ivan, I cannot ask questions because I will be responsible for answers.
Yes, that's what I will do. That was my plan.
I cannot, sorry. I have some questions, but so thank you.
Yeah, okay, I'm stopping. I stop sharing them.
Thank you very much. And again, we can use these.
Thank you.
Hello.
Hi,
Unmuted.
Andre.
Hi, hi. How are you?
Good, good. How are you?
Okay,
The
we
virtual
are kind
coffee.
of face. Yes, sorry.
No, I was saying the virtual coffee didn't work, so I had to grab a real one. Okay.
So thanks again for accepting contribute to this workshop.
I mean, thanks for
The
the
second part
invitation.
will, yeah, the second part will be more kind of oriented towards applied things.
Yeah, I mean, it's really a nice workshop, you know, like, I mean, I didn't have the time to process all, so I'm really glad that people will share slides they will record. But yeah, you know,
It's
interesting.
but you're not against recording your presentation? Or
No, I'm fine. Yeah, it's fine.
If you're fine. We have our group YouTube channel, and we are going to host the whole meeting at YouTube channel. So the slides will be there also. Okay, I suggest that we wait another 1 minute, but you can already start sharing your screen.
Okay. Let me try. I usually use Windows.
Yeah, I can see it.
Okay.
Cool. Okay, welcome back, everybody. We start our second part of this workshop on complexity and simplicity in real-life data sets. So I'm very glad to present Luca Pinello, who is an associate professor at Massachusetts General Hospital and also at Harvard Medical School. So Luca received his PhD in computer science and mathematics from University of Palermo in Italy, then moved as a postdoc to work with Professor Yuan at Dana-Farber Cancer Institute, where he studied the role of chromatin in gene regulation by computational methods. So afterwards, Luca started his own lab, and the main interest of his lab is innovative computational approaches, cutting-edge experimental assays to systematically analyze sources of genetic and epigenetic variation. And the lab uses very extensively machine learning approaches, data mining, high-performance computing to solve computationally challenging and big data problems. So please, Luca, the stage is yours. 35 minutes, and if you can leave a couple of minutes for questions, would be great. Thank you.
Yeah, absolutely. Thanks a lot for the invitation. It's a pleasure to be. I'm enjoying a lot, you know, the workshop so far. So briefly, my lab is trying to study gene regulation, and today I want to present this method called Simba that essentially try to create embeddings where we embed, in addition to cells, we also embed features. And this collaborative work was led by Yidong Chen, that was a postdoc in my lab, and Adam Lehrer is from Facebook. You know, they develop a nice graph embedding technique, so we collaborated with them, and then with two PhD students in my lab, Jay Yang and Michael. So my lab, as Andre mentioned, we are interested in applying machine learning to recent biological assays, one for CRISPR genome editing and one for single cell omics. And you know, the general question is like we want to understand which part of the genome is functional, and also we want to understand how the cell make their decision in development, and try to understand the gene regulatory circuits in the cells. So in order to do that, you can approach this from multiple point of view. And one thing that we are really excited are single cell assays. So I don't know what is your biological background, but gene regulation is really complex process. So you have, the genome, you have like genes that can be expressed in different cell types and at different time. And there are a lot of, mechanisms that regulate gene expression. And, there are like some, that I will talk. So I just want to spend 1 minute to introduce. So one is like the conformation of the DNA of the chromatin. So the idea is that certain region may be more open or closed, and the region that are more open are important to gene regulation because they create some kind of loops that promote gene expression. And you can measure also many other things in the cell, and like there was an explosion of assays to measure the single cell level in addition to gene expression level through messenger RNA, also these other quantities. So from high level point of view, what we're trying to do is like you take a cell and you encode this cell as a set of features, and you can create different matrices for the features that you can measure in a cell. And as we learned from the previous talk, this is the classic example where the number of features is much more than the number of cells that you profile. So all the I really enjoyed the previous talk. I think there are a lot of potential application for that framework. But the goal is like you have this set of measurements, and then based on this set of measurements, you want to understand like how the cells are controlling gene expression of the different cell types are organized. So another high level picture slide. So the goal for us is like starting from these measurements, try to create embeddings and visualization that highlight principles of gene regulation. And we call this thing gene regulatory maps. And for people that are more familiar with the single cell modeling, usually people have proposed to use UMAP and like reducing dimension based on gene expression. However, in this map, usually you have only cells. So this different color here represents cells, but in addition, in these maps with Simba, we're trying to embed also the features. For example, you can you can have a vector that tells you like there is a particular gene that is close to a population, and maybe this has a particular meaning for that population. And Simba was inspired by methods that people have used in natural language processing. I will motivate why we started to explore these methods. Like as many of like natural language processing is just a way to teach machine to understand and model text and words, and there are so many different applications of NLP and related methods. And there are like some that are really relevant for biology, like information retrieval, text summarization, and also here you see like social networks. So social networks are really powerful way to encode relation, and I will show you like this is like what we're trying to do with Simba, and this approach seems to be really fruitful. So, but if you think about natural language processing, if you have text or like natural text, it's kind of simple. We know like how to tokenize a text, and we have a sentence, we break in token, and then in some cases, for example, in social network or Twitter, you have like some metadata that can be like hashtags. In biology, like what are the words? Like what are the sentences? So there are all these questions that we need to think about. So the analogy is like more a cartoonish way. So can we create some kind of chromatin tweets? So imagine you have like different genomic tracks, and when you have like these peaks here, these are the regions that are more open in the cell, that are more functional. So what about if we like take the DNA sequence of these peaks and just tokenize with some k-mers, and then maybe there are also other features that we have in this region, and then use the hashtag as cells. So this is just one way that we potentially approach this problem and apply all these NLP techniques that people have developed. So just before, we try to formalize this, just really high level, some concepts. So, in word embeddings, I think, is really important. How do you embed words, right? So these are discrete objects. So the idea is that, we want to find a space where words that have similar context would be closed in this space. So the way you can do this, you can think as a bag of features. You take each word in a sentence and look around, and you find this bag of features. And then the task is can you find vectors that will put together words that have similar context, and you get this kind of embeddings. And the nice thing about this embedding is in addition to group words that have similar meaning, you can also find additional properties of the space. For example, you may have a particular vector that has some particular meaning. For example, the distance between man and woman is similar to the king and queen. Or for example, another property is if you have this embedding of city and capitals, you can find a vector that given a country would find the capital. So just really powerful and surprising properties that emerge. And so the way to do this, there are different ways. Some of these are solved with small neural networks, but the input again is these words, and the output is what they call word embedding. So you encode this word with vectors, and then just to visualize and to please our eyes, we can embed in 2D or in 3D, and then we can start to again try to assess some meaning of this word using vector operation and proximity. For example, another thing, a classic example. How do you get a queen? To get a queen, you can take a king, and then you add, the woman minus the man, and you get the queen. So there are all these things that are really interesting. And the field, since this initial word embedded, word to vector, and other methods has evolved, dramatically. One thing that we were really excited while we were exploring these NLP techniques is this method developed from Facebook called Star Space. And they describe different way to embed not just words, but also to embed hierarchical structure. So imagine that you have an article, and then the article can be decomposed in sentences, and these sentences can be decomposed in words. So what they propose is can we find an embedding where you just don't have the embedding on the words and then operate on the words, but you can find actually a vector for every node in this hierarchy. So, and the idea is you start to get into these graphs, right? So now you have this hierarchical structure, and you find these spaces, and then the property of these spaces, you can using just cosine distance or dot product, you can essentially assess the similarity of these things are related in these spaces. And we were really excited, so we started to explore this this concept. And then the question is okay, we have this nice model, and in biology things are for sure hierarchical. So do we encode all the things that we know and our understanding in this graph? So our biological sentences should account for different molecular features. And for people that are not familiar with gene regulation, so the concept are you have your cells. These cells can express different genes. So in the genome there are region where you have your gene, the coding region, and this can be transcribed. And there are a bunch of proteins that goes nearby this region. They can bind the promoter, and then there are this distal region called enhancer that can promote expression. And these are what you can profile with what I call chromatin accessibility. These assay that find these peaks. And then this region you can find sequences that can be used to understand which factor can bind. These are called activator, transcription factor activator, or repressor. So these are the actors of our graph. So now let's think about how can we encode this in a graph and then, try to embed this graph such that all these, nodes, in this graph would be in the same space. And then we can try to study like how they are related to each other and try to understand based on proximity, if there are like some biological properties that we can study. So this is like, with this concept in mind, let's just, go through the workflow that we propose. So the first step is like, you need to do an experiment. So you profile single cells and you measure, gene expression, chromatin accessibility, these sequences that, essentially are a way to find which factor will bind, and k-mers that, just, a way to just capture things that we don't know yet. So then, the second step is like, we can, encode this in a knowledge graph. And, here, the graph that we will propose are, the following. So you have, your cells, and then we have, edges to say, for example, that a cell expresses a gene, and then, a cell may contain a peak, and, a peak may contain DNA sequences. I will show you how we embed this using a graph embedding technique that is a extension of star space. And then finally we will find a space after this graph embedding that have some properties that I will show you that are nice to answer important biological questions. So let's start from the encoding. So what do we do? So we essentially encode our observation as edges. So these are the single cells that we measure, and every time, for example, if this cell in the matrix, in the entry in the matrix express a gene, we add an edge. If a cell, has a peak, we add, an edge, and so on. If a peak has a particular transcription factor or motif, these are interchangeable words, we add an edge, and so on. So each edge is an observation. And, we propose this hierarchical structure that capture our understanding of how these things are related. So now we need to, take this graph and we want to embed this graph. So to do this, we use this PyTorch big graph. And, the output of this is like a 50-dimensional space where, each node here that can be a different entity would be like a point in this 50-dimensional space. So now, like, what is PyTorch big graph? So it's like a really nice library that, Adam Lehrer at Facebook developed. And, they can find embedding for, graph with billions of nodes. I mean, they are Facebook, they have, a huge graph. So they spend, a lot of time trying to, optimize this problem. And, importantly, they support multi-entity. So in our case, these different nodes and multi-relation graphs. And, there are a lot, you can customize this, in many different ways. So let's go through, this, the input and the output, and, what is the methodology. So the graph, is like, this is like the graph, the pictorial thing. So you have to specify what are the relations. So the relation essentially are the set of edges that connect two particular entities. For example, cell and gene, or cell and peaks, or peaks and k-mers. Now that you have a graph, how do we embed this graph? Again, this is like discrete structure. So let's say that we have a graph G where we have V that are the nodes, R is like a set of relations, and E is like a set of edges. So for each edge, you can encode this as three like source, the relation, and the destination that the edge has. So the first step to find this embedding is like, first, you need to define a scoring function. And a scoring function essentially is a function that takes in input one edge and give you a number. And the way they do this, you can do in so many different ways. The simplest way to get a score for an edge is like through the dot product of the encoding. So imagine this is like, your vector encoding the source, and this is the vector encoding the destination. And G can be like, diagonal matrix. So you don't do anything. So you just essentially the vector, and the similarity can be cosine or dot product. in general, this can be like, you can also have, we have this G because, this can be an affine transformation. So you can, apply another function instead of, just using like directly the dot product or the cosine. But, this is like a function that, give an edge and give you a number. So what about the loss function, right? Whether we tried, how do we optimize, this embedding? So actually it's quite simple. So what you do here, you are trying to maximize the separation between the scores of, the edges that you observe. These are the E, and the edges that are not possible. So what they call negative edges. And then there's like, this lambda is kind of, in subvector machine where you have like this margin, so you have some tolerance. And then, you go over all the edges in G, all the edges in this, S prime E. So these are, the negative edges. And then, you optimize this function with gradient descent, and you find the embedding. So it's quite simple, approach. There are a lot of things to make this scalable and, to scale to a number of nodes. And, we work with Adam to make sure that this is applicable to single cell data. But this is essentially the method, really simple method. So in Simba, we made some modification. So instead of using this margin, score that, it's good, if you want to have a ranking of, the edges, we use a softmax loss because, we want to have for each edge, some kind of probability that, for example, a cell is connected to a gene. So the softmax, is a better way to do that. And then, one thing, problem with dimensionality, the number of, features is way larger than the number of cells. Given that here we are embedding everything in the same space, the feature will dominate. And this is a problem for the embedding. So we actually have different weights in this loss function to scale, proportionally, depending on the number of features, the contribution to the loss function. Then, we had to add a regularizer. And then, we assess convergent of overfitting with cross-validation. So this is the method. So after this, you have, your graph embedding. However, it's still not what we want. And, the reason for that is that in this space, similar to, this slide I show you here, you will see like, for example, all the cells and all the genes and all the peaks would be in different parts of the space. They're organized in a way that, the model would predict, the probability to be connected based on dot products. However, we want something, in which like the cells and the genes, they are like close to each other if they are like, related. So we play another trick here. So from this graph embedding to get the Simba space, what we do is like we select an entity type that we are most interested. For example, the cells, right? Like we want to understand what is relevant for a cell. And then, we take, an entity that we want to like project in the same coordinate system. And then what we do, like, imagine this is your probability of, a particular gene. So imagine this is square is the gene, the probability over all the cells. And it's just a softmax. So, you can think as a probability. And then we just take the weighted average of the coordinates of all the cells that are, potentially connected to this gene. And, we find a location in this space. And, this location actually is really important because, in this way we can actually compare the genes to the cell. And, the interpretation is like, this is the weighted average of the coordinates of the cells that are relevant for the gene. And, this is like another interpretation because they are like weighted based on the edge probability. So this is like the Simba space. So finally we have this space, like, why, we care, about doing all of this. So because, now you can, start to answer a lot of questions about, can we find relevant subpopulations? So we can cluster, for example, the cells. But now that, the genes are in the same coordinate system, we can find genes that are nearby these cells. And these maybe are important genes for those cells. So we can find marker genes. And then, you know, like, given that we also embed these motifs that are, you know, this sequence that, you know, we use to model transcription factor binding, we can actually ask, what are, you know, the transcription factors that are relevant for these cell types? And finally, we can start to uncover principle of regulatory networks, you know, if we start to study the proximity of all these different entities. So I will show you a few examples of this. So importantly, like, you know, with this approach here, you know, we show that you can, with the same essentially modeling, you can conquer all the single cell tasks that people have, you know, proposed so far using essentially the same framework. So you don't need to create specialized methods to, for example, do batch correction or multi-omic integration. So you can essentially use the same technique and cover, you know, all the tasks. And, you know, I believe this is like, you know, quite nice, you know, result, because, you know, from, you just need to decide what are the rules to encode this graph. And then, you know, like you can essentially get this for free. So let me show you the, you know, few vignettes just to understand how this thing works. So if you remember from my first slide, you have, you know, single cell measurement. So this matrix looks like this. So you have, you know, the rows are the cells and the columns are the genes, or, you know, some people, they just rotate. So what you need to do, the first thing we do is like to discretize this distribution, because, you know, we have edges with different weights, and, you know, just denoise a little bit the system. So we discretize the expression, and then we have different edges with different weights. And the graph here is quite simple. You have like your cells and you have your genes. So you take this graph, you embed, you know, just to give you a real example. So if you embed that you just look the cells, you can essentially conquer clustering. And this is what, you know, everyone in the field is doing. You take your, you know, low dimensional embedding, you cluster the cells, and you find the different populations. However, in Simba, you create these spaces where, you know, you can, in addition to these cell types, you can actually plot the genes. And then, you know, one interesting property of these spaces is that the genes that are close to cells are genes that are important for that population. Genes that are not important for the cells, the method will embed, you know, in the middle of nowhere. So for example, these two genes here, GAPDH, you know, this is like a skipping gene, is expressed in all the cells. So it's not really cell type specific. It's not close to any cell group. Whereas, you know, this gene here, you know, is really important for B cell, and, you know, you can really see that it's pointing here. So it just, you know, like highlighting this particular location. And the beauty of this is that you don't need to do clustering. You know, currently people, they do clusters, and then they compare clusters to find relevant genes, but the clustering resolution really limits you, because, you know, like, imagine you don't know what are the cell types, you know, that's what we want to do. So if I will remove the colors, you can say, well, you know, maybe I have 3 clusters, maybe I have 4, maybe I have 5. So it's really limiting, you know, this approach. But we don't need to do any clustering. And then if you look, you know, this softmax probability, that is just, you know, a way to essentially tell, you know, for example, this gene, what is the probability to be assigned, for example, to this population, to this other you see, like, it's really skewed, right? Whereas, you know, our skipping gene is like kind of boring, you know, almost uniform. So then, you know, we reason, like, instead of doing clustering and then find marker genes or look in this map by eye, what about we propose some measure, you know, matrix for skewness, right? So that's what we did. You know, you can, for example, take, you know, the top and, you know, genes over the others. So, you know, using the Gini index, that, you know, is a nice matrix of imbalance. And with this, you know, trick, you can actually discover all the marker genes that, you know, people have described before, but, you know, also other one. And, you know, whereas, you know, like skipping genes will have really low matrix score. So we have, you know, a way to find, you know, important features in this space. So let me show you now, like, a toxic, you know, this is like, you know, say where you profile open chromatin, or, you know, these peaks I show you like multiple times. So the matrix here looks like this. So you have the cells, And for each genomic region, you have like either 0, 1, or 2. So these are, really, sparse matrices. And then, if you have this, it's really similar to the previous task. So you have like the cells, and then you connect the peaks. However, these are regions in the genome. So you can actually ask, can you encode also the presence of, particular DNA sequences? So you can enrich this graph with another layer. So that's essentially what we do here, really simple operation. And we show that, if you do this graph, you can, if we start from the embedding of just cells, and if you do like what everyone is doing right now, clustering these to discover population, so you can get, performance as state-of-the-art model, in some cases even better. So we published a benchmark paper last year, and these are all the methods. So we had Simba, and we show that actually if you do this graph embedding, you get state-of-the-art performances. And then in addition, here you can select, in addition to the peaks, also DNA sequences and motif. And the nice thing is like this new visualization, these regulatory maps, where in addition to the cells, now we can also project all the other features. And then this is nice because you can start to explore neighborhood in this space, based on the gene index, or just literally you mark one area and you ask what's going on for this red population that is like red blood cells. So you can say, well, I see that there are some peaks embedded here. I see that there is like, some transcription factor. So, and then, you can actually go in the old school, visualization, look in genomic tracks. And these are the different populations. And then, for the red population that, is likes to be in this region, the space, you see that there are like specific signal as expected. So, the embedding worked really well. And also, these peaks contain specific DNA sequences. So this is quite nice because you don't need to do all of this and, do, all this enrichment operation, but you just look for proximity of, these embeddings, embedded vectors. And you can discover, things that are relevant for that cell population. And again, here we can use, this barcode plot to show that these are actually extremely skewed features. And that's why, they are embedded in this location. And we can also validate this, using orthogonal methods, for example, to show that GATA1 indeed is expressed in this erythroid population, and this chimer that, really match well this GATA1 binding site is also in that region. So the last, I have, a few minutes left, so I will just talk about the last application. So this is like the most exciting to me. So in single cell data assays, now in addition to profile gene expression or chromatin accessibility separately, you can profile together. So in one cell, you profile gene expression and peaks. So you have, now both these matrices, and now you can start to create this more rich graph where you have like everything, everything that, I just showed you before, but now like it's all in, one cell get all these measurements. And then, we try to embed these things. And here you see like, similar to before, how the space, get richer and richer. So if you just profile cells, and this is like mouse skin development, you can cluster this and you can even do trajectories. And, this is like a nice space. However, when you start to look for genes, you can start to embed genes in the right location to mark, the right biological population. And then when you start to put everything together, you get these beautiful regulatory maps where now you have like all these entities, in this space. And then you can start to invent new ways to, extract insights. One thing that, we play with is like, can we recover regulatory circuits from this thing? So, one game that you can play is that, imagine you have like a transcription factor DNA sequence, motif, but here you have also the gene expressing this particular transcription factor. So then you can say, well, can I look, can I rank all my transcription factors based on this distance. So I would put on top things that, you know, where these 2 points are close to each other, so they're more consistent. So we do this, and, you know, what you find is like quite nice that, you know, you find all the master regulators. So, you know, this left one, you know, is like something that, you know, in this paper where we got this data, we're really excited. So these are the genes that are actually driving differentiation, and we have a recipe just based on proximity to find them, right? So, you know, just, you know, like really nice result. And then, you know, we reason, what about, you know, like, you know, the genes that this factor are regulating? So, well, you know, like if you have already these master regulators, you can actually look nearby, and then you can ask, you know, if I am a target gene, I want to be like close, you know, to these embeddings, but as well, you know, it would be nice, you know, if there are like peaks for this gene that contain, you know, this transcription factor binding site. I know it's really convoluted, but, you know, these are essentially, we're trying to, you know, put all our knowledge here, but, you know, just using proximity. We don't use any other operation. So if you do this, you know, like proximity game, you can find these regulons. So now you have a way to find a transcription factor and also to map, you know, genes that, you know, this factor may regulate. And, you know, again, all of this is based on proximity. So I will stop here because, you know, we're already at 30 minutes. I just want to say we, you know, posted the preprint on Barchive describing, you know, all the details and, you know, show like how to use, you know, Simba and Pradis. We have, you know, also GitHub is, of course, is open source. And also we spend a lot of time to create a documentation website so you can learn, you know, like how to use Simba, you know, if you're interested. So in conclusion, you know, I was really excited to explore NLP techniques, and, you know, like as I hope I show you, like you can apply these techniques not just to text but also to biological measurements. We can encode our single cell measurement in hierarchical graphs, and we can embed this graph, you know, in the same space together with the defining molecular features. And also we can do a lot of things just using proximity queries. You know, we call these biological queries. And, you know, these proximity queries can uncover principle of gene regulations. So, I mean, there is still a lot of work to do, you know, in this area, I believe. You know, this is just one, you know, embedding method, but we are excited to explore more. So if you have other ideas about, you know, if you're working in this area, I would love to chat. And also, you know, we're thinking about, sometimes, you know, biological processes are dynamic. What about if you have time points? How do you encode these time points in these graphs? What about if you have like perturbation of nodes, for example, through genome editing? And, you know, maybe there are other operations that we can define in these spaces. And yeah, so these are, you know, some future plans for this area. And with this, I would like to stop, acknowledge Idon Chen that, you know, was leading this project, Adam Lehrer from Facebook, you know, really helped us a lot to learn about these techniques and to adopt PyTorch Big Graph, and Jayang and Michael for, you know, helping us with this project, and everyone in my lab. And I will stop here. I will take questions. Thank you.
So thank you very much. It was super interesting. So are there any questions from the audience? So we have a question from Evgeny Mirkis, please.
I have only one big question. In all your graphs, probability of linking was less than 1%, the highest. How you interpret it?
So, sorry, so again, in my graph, the probability of?
Probability always was less than 1%, the highest probability.
Oh. Yeah. I mean, this is like, you're referring to this, right?
No,
so,
for any, yes. For example, this.
Yeah. So, you know, this is just a property of the softmax, right? Because, you know, these things over all the nodes should sum to 1, and, you know, we have like so many nodes in this graph, right? So it's not, you know, like the, you know, that you need to expect a high probability like for few nodes, but more like, you know, the relative distribution. So It's not a problem if, the probability is really low, because, like you have so many different nodes, and again, you have this constraint that should sum to 1. So, again, the property is like you want to see this nice skewness, right, for few nodes. Yeah, that's a good question.
And one more question about embeddings. You use, I completely understand how you define positive edges, but I don't understand how you define negative edges. Yes,
that's another good question. I mean, like, yeah, you're hitting, good points. So actually in the paper, the original PyTorch bgraph paper, they have like a large section about this. So do you like find negative edges? So the operation they call corrupting the graph. You need to corrupt edges. So what you do, like, to build this set of negative samples, you fix the destination or you fix the source, and then you need to sample, so if you fix the destination, you will sample the source by respecting the relationship. And now you can sample in 2 ways. One is like you just sample randomly from the set of, the source node, but this may kind of dilute your power because you can sample, some true, observed edges. So this is what they call uniform sampling. So you just sample, nodes fixing each destination. The other way is like actually respecting, like your positive. So you can say, I can sample an edge, I can sample a source node, but this source node should not be present in your, in your observed edges. So these are the 2 ways. And, the first one that is uniform, and that's what they use, is really fast because you don't need to check anything. It's really appropriate for really large graphs. The second one is better if you have like, more smaller graph, because, you don't want to dilute your signal, right? Putting, back some edges that are positive as negative. So that's essentially what they do to find, to create these negative edges so you can actually train, this model.
Thank you.
Yeah, of course.
So I guess the second question was from Alexander Gorban, please.
Hi, Luca. It's nice to see you almost in person.
Yes, it's really nice.
So this is, I have a rather difficult question for this for me.
Okay.
The points in your vertex graph you call cells, but roughly speaking, everything is we assume that in this picture that the cells are stationary, and the state of gene activity is a property of a cell, but the cell during its life goes through different situations, through different chemical environment, even different stage of cell cycle, and many other changes. Cell is not stationary. Therefore, and this cell and these fluctuations could be, I don't know, this is my question. These fluctuations in the state of the same cell may be bigger than the difference between different cells somehow in amplitude. Could you please comment this point? I just don't know. You know this better.
Yeah, no, I think it's a really good point. We're assuming that if the cell, they don't change much. This the whatever is the cell state doesn't change much. Probably these representation are interpretable. But however, if you have like a dynamic process where maybe like immune response where we capture the cell in that moment, but actually the cell was evolving toward other states. So when we do this, in this graph, what will happen is like you will see like maybe some kind of dispersion, hopefully through the developmental axis, right? So that's my intuition. What will it's like in this you kind of see like a little bit this phenomena. So in this system we know that this is like a differentiation system. These cells kind of go in this direction. So what happens is like you don't get cluster, but you get this continuous graph. However, like this is not optimal, right? Like it's not optimal because in this graph, like everything is kind of fixed. So it would be nice to think about dynamical graph, right? But I don't know yet, to model this. But and that's why I had this. How can we encode time points or dynamic here? Because right now we assume that everything is fixed. So as you say, like if this change, if you will observe, the cell in multiple points may confound, this interpretation. The problem is that single cell measurements are destructive. So when you measure a cell, the cell is gone. So you cannot really track the cell. However, the one thing I didn't mention today, there are like techniques called lineage tracing. So you cannot track a cell because when you measure the cell is gone, but you can actually learn like that one cell is coming from another cell. So in this graph potential you can say I observe this cell, I destroy this cell, but this new cell is coming from that cell. So you have this kind of constraints in a way that we can use to like adjust a little bit this representation. But I mean, we're not yet there. But if you have other ideas of to like explore other more, I would love to chat. But yeah, the summary is like you may get still something meaningful, like if you have like development and if you have these processes, but this doesn't guarantee that this will work always, right? So that's why more powerful models that takes into account the dynamic when they embed would be more suitable. But I don't have yet these models.
Thank you. Thank you.
Yeah, of course.
Thank you, Alexander. I think we had a question from another Alexander, from Alexander Chervov. Please.
Maria Comparator.
Thank you very much for very interesting talk, Luca. So my question is, if we consider just NLP, natural language approach, we can find, for example, take some trained model like BERT and find some name of the genes, and names of some other things, and what we obtain, a kind of, then we can connect them, for example, by the KNN graph. And so we'll obtain some graph which contains different things. No, it's not the same as yours, but conceptually it's genes, for example, you can put drugs, you can put diseases, and so on and so forth. So do you think this will be somehow related to what you're doing?
Yeah, actually, yeah, I think it is related. Like this is another area we would like to explore. I was watching a talk from the author of UMAP, Leland, I don't remember his last name, but essentially he's exploring, these models. He has his own model, but he was talking about BERT and, like, using, like similar, I mean, he has his own, I don't remember his name, but I think, it's really interesting, idea and areas. So, I don't know enough about these models to like have a recipe, but your intuition is like you will use BERT, you will embed different things, and then you use the KNN, like can you encode with BERT, hierarchical things, right? Something like this. So what will you do in that case? Will you, do the encoding, independently? Like this, I'm not, like super I know the output, but in my mind, I don't have a solution. I think it's a really interesting area, but I don't know. We'll approach this hierarchical structure with these models.
Well, no, I haven't thought about hierarchical structure in that type. Yes, but yeah, I didn't mean that it's exactly the same. Just but kind of, for example, we cannot speak about in the natural language. We cannot speak about the cells like in single cell, but we can speak about the cell types, yes, because you can analyze literature, take different cell types, names in the literature, you can find them, and then you can kind of construct a graph which will contain cell types, for example, drugs, diseases, and so on and so forth.
I see. Yeah, no, it's really interesting. Yeah, we need to explore more this area. Yeah. Thanks for the suggestion.
Okay, and we have the last question from Maria Kondratova.
Hi, Luca. Thank you very much. I have two questions. One very pragmatic. Is it how user-friendly? I'm biologist, so my first question for any program is how user-friendly is your program and where could we use it? Is it web interface or it's R Python package? And the second question is, I put it all together. And the second question is, did you apply your method for bulk data also? And what do you think about difference between, because there is essential difference between bulk and single cell data in many terms, but methodologically, I do not see any reason why your methods could not be applied for bulk data classifications. But what do you think about it?
Yeah, these are excellent questions. So for the first question, like if you take a screenshot of this slide, you will have especially this website. I think it's quite user friendly, and it's written in Python, and then we have like Jupyter Notebook, that essentially you can just change a few lines, put your data.
Okay, I have unstable internet connection. I have missed this slide. Okay, great.
So yeah, I think and if you have questions, please shoot me an email. We are happy to help. So you can get started with this. So for the second question, like we never try for bulk measurement. So my intuition is that it may work well. However, this graph would be like really small. Like in the sense that like one part of the graph would be really small. So if you think about, this kind of graph, imagine that you have like, bulk ataxic data. So in which you have peaks, the peaks would be still 1,000, but the number of samples would be really small because instead of profiling, thousands of cells, if you have bulk measurement, you may profile maybe 3 or 4 different samples. So I think, would work
more about transcriptomic data. Yes, attack sequences.
Yes, I think if you
I thought
think
more
about
this, huge transcriptomic data sets.
Yes. So in that case, imagine you have like thousands of genes, right? So what you will see, like imagine these are your condition. You may have like biological replicates. So the number of samples, will not be really large, right? The biological sample. You may have like 10, 20. This layer here will not change. It will be exactly the same because it will be essentially thousands of genes. But this part here will be like really small. So when you will do these embeddings, you can still do something like this. But instead of having like multiple points for the cell types, you will have single dots, right? So I think you can use Simba in that configuration. And like the question is like if you have a single assay, like what can you learn with this method? Because this is like nice because if you have heterogeneity within one population, you can learn that. So I think it would be applicable. You can still use all these metrics that we discuss. On the other hand, I feel like more tools like DSIC2 or other tools designed for finding relevant genes maybe like are easier to adopt in that case. So,
okay, thank you very much.
Yeah, of course.
Thank you. We will try to apply and I will be glad to contact you if I have questions. Thank you.
Absolutely. Thank you.
So thank you. Okay, again. And again, virtual uploads. So now I guess it's my turn. I will try to share my screen.
Yes. So yes, Andrei, and whilst you're uploading the screen, if I may, I would like to introduce you to the audience. So, let me take this opportunity and just a bit of time to make the introduction. So, Andrei is a group leader of computational systems biology of cancer at Institute Curie, and he also is a chair at Paris Artificial Intelligence Research Institute. This is for those who are in the UK, this is a twin brother of the Turing Institute, established by the UK government. He's also co-author and author of more than 100 publications at the interface and intersection of machine learning, mathematical modeling, and cancer biology. And in particular, he co-authored a book on principal manifolds in data analysis. And I think also he is an author of the first textbook on cancer systems biology. So I would like to give the floor to Andrei now, and now he'll talk about elastic principal graphs for modeling complex multi-dimensional data and their application in biology. Andrei, the mic is yours.
Okay, so thank you very much. Everything is okay with the screen, and can you see my mouse just in case?
Yes.
Okay, great. So thank you very much for introduction, Ivan. I'm going to talk about methods for exploring complexity of data. Many of the people who are present here in the audience, they contributed to this work, but I decided, so my decision was not to put them as co-authors, even if they are true co-authors of this work, just not to prevent from asking questions like like in the previous talk. So please don't hesitate. So just to set some common ground, we are going to talk about multidimensional data geometry. So we have a data point cloud in multidimensional space. It can be labeled or not necessary. We will try to avoid talking about existence of underlying probability distribution. Well, yeah, so we leave apart the question, does it kind of exist or not, or can it be measured or not? We just will try to talk without referring to this notion. And in the classical geometrical approach for data analysis, we have these standard questions of what is the right data matrix, what is a complex or simple data geometry, what does it mean, or topology, structure, and data manifold hypothesis, which is very common in this field. But my first point, and I'm not that much original here, and actually it was already said in one of the answers to the question, one of the questions actually, is that looking at real-life multidimensional data sets as uniform samples from a low-dimensional manifold is a very limited point of view in data analysis. And the real-life data sets are far from being uniformly distributed. They are structured, and they are structured in many different ways. Well, the simplest deviation from this uniform hypothesis is that the data can contain clusters. But here you can see a small gallery of UMAP representations of different data sets from many different fields. In this case, they are ordered by estimated intrinsic dimensionality from low dimensional data sets to higher dimensional data sets. And you can see that here something interesting is happening. So at first, these data sets look kind of might look simple. Then they become more complex, and then they become simple again in high dimensions. So this is a reflection of some of the things that we discuss here at this workshop. And of course, the big question is how to develop language to describe all these types of complexity, or types of structures, type of organization of real-life data. I'm not ready to give you kind of even a sketch of such language. It must be a subject of very long-term research program. Just we are going to discuss one particular possible element of such language. And one of the notion of the notion by which we describe complexity of real-life data sets is the notion of cluster. And let's say, theoretically speaking, the mathematical notion which underlies this notion of cluster is a peak of data point density. But just like is in real motions, we might have peaks, but we might have also something which is called ridges, so some kind of things with which we might connect peaks, or just they can exist as a ridge. So this is another element which might shape complexity of real-life data sets, and I'm not sure that there is a standard name for indicating the presence of ridges in the data, in the density. Sometimes they are called filaments, sometimes they are called fibers in the data. And if you have such things in the density distribution, then you have a quite distinct organization of data in multidimensional space compared to cluster-based organization. So the notion of region, the density region, the data can be even formalized, and there is even an asymptotic theory of these density regions. So they can be formalized as using some eigenvector decomposition of Hessian of the density function. But as usual with density functions, I mean, to apply this theory, you have to quantify density, and this is not an evident task. So I mean, this kind of theory is applicable as usual in a relatively low dimensional space. In real life data sets, there are some data sets which are famous for having ridges, density ridges, and probably one of the most famous is this cosmic web, which represents a distribution of galaxies and maybe the velocity vectors in the observable universe. So the gravitational forces, they shape filaments. They shape filaments consisting of galaxies, and some people develop methods for extracting these filaments from complex distribution of these galaxies. But what we like more in our group, and we maybe the richest data sets characterized by density ridges today are from completely opposite scale, and they represent the same unicellular measurements which were discussed in the previous talk. So Luca has nicely introduced this area, and in this area there is really kind of technological revolution which produces a lot of interest to analyze multidimensional data, and it helps us to understand biology in the end. So each cell is characterized by multidimensional vector, and individual cells can undergo differentiation following a series of bifurcations, with Waddington landscape being the most popular metaphor of this process. If we think that there exists some kind of at least local Lyapunov functions which kind of which determines the local minimums of some potential in this multidimensional space, we can think that indeed the density of points in multidimensional space are characterized by ridges which represent kind of a mirror of this Waddington landscape. So Luca has already presented you some examples of single cell visualizations. Here yet another one. It's a single cell data cartography of planarian, which is the simplest worm characterized by remarkable potential for self-regeneration. And if you can cut this worm into parts, and it can regenerate into adult organisms. So this is because almost half of the cells in this worm are composed of stem cells. So you have constant process of differentiation. It's actively differentiating cells in this organism. So in the end, you can take this worm, decompose it into individual cells, and if you will project this multidimensional distribution data point cloud into low dimensional space, you will clearly see these roots of differentiation, and you can talk about them as density ridges in the data. So the question is, can we derive a data model which captures this complexity at necessarily scale? By the way, this single cell approach and kind of looking for trajectories in single cell data can be scaled, can be at least imagined to scale up at the level of human organism. And currently there are multiple efforts having in mind that at some point we will be able to have access to the molecular state of each cell in a human organism. At least we'll have an idea about trajectories which each of the cells in the human organism follow in, let's say, healthy aging scenario, which is represented here by nice with a blue color. And also, we will be able to characterize bifurcations which lead to deviations from healthy aging trajectory and lead to some pathological cases. So the idea, the hopes that this single cell technology will allow us to detect such bifurcations relatively early before appearance of any symptoms, where the application of therapy will be the most efficient. So few more words why we think that the density regions sometimes represent trajectories. It's because we think of, for example, individual cells as objects that have been captured in a single snapshot in the active dynamical process at different stages of it. And each object is characterized by some state in multidimensional space. And the states form a sequence of data points, and they are ordered in physical time. But when we observe the snapshot, we do not always know the real-time label. We only observe the sequence of states. And if you can approximate in multidimensional space the sequence of states by, for example, by this curve, then we can quantify, we can introduce a notion of pseudo-time, which will allow us to measure the progress of all these objects along the dynamical process, starting from hypothetical root to the end of the process. It's not always feasible as an approach, but if you observe actively developing dynamical process, this might be possible. And in applied statistics, there are well-known methods how to approximate these curvilinear data point clouds. And they start from the works of Hastie in the end of 80s, where he suggested that sometimes if you have non-linearity in the data, the first principal component is not the best approximator of the data, so you have to introduce something which will be non-linear and will pass through the middle of the data distribution, serving to approximate it. And this something which was called principal curve should possess some regular properties. For example, this particular curve, it has very good approximation properties in the sense that it stays very close to any particular data point, but probably, I mean, the assumption is that this construction doesn't help to generalize well, so it doesn't help to predict where new data points will be. So this is why it's important all these regularizers and smoothers of principal curves that you must introduce. And we also introduced some years ago. We suggested an efficient computational method to construct this principal curve. They're called one-dimensional elastic maps. They can be also two-dimensional, and the idea is that you use a system of springs, which at the same time they attract nodes of some kind of linear graph to the data points. And there are springs which attract nodes of the linear graph to each other. And at the same time, you have negative springs which repulse nodes of the approximating graph, one through one. And if you will try to minimize the elastic energy of the system of springs, it provides you a smooth and regular curvilinear approximation of your data point cloud. But here there is one complexity which is might be quite common when you observe real-life dynamical processes, including those which are involved in differentiation, is that sometimes the dynamical process is characterized by a bifurcation. In this case, you start to have branching snapshots of the data point distribution. And then you wish to approximate this branching pattern with a set of principal curves. And moreover, these principal curves might have some common parts at which they coincide, and then they diverge accordingly to the underlying bifurcations. So to deal with this situation and to introduce a mathematical object which is able to kind of capture this complexity in real-life data point clouds. Some years ago, almost 15 years ago, we suggested these principal trees, which are tree-like approximators of data point clouds. And more generally, we suggested a framework of elastic principal graphs, and this is something that I will describe in my talk. So the notion of principal graph was coined by Balázs Kegel in 2002, first in the task of skeletonization of handwritten digits. But then it was converted to general purpose machine learning methods, which can approximate not only two-dimensional data point distributions, but it can work in a multidimensional data space as well. So today, I mean, this method is described in multiple articles, in several books. It's implemented in several programming languages, including MATLAB, R, Python, Scala, Java. So it exists and it can be used readily in many practical projects. So there are two things which distinguish this elastic principal graph approach from other similar methods which try to solve a similar task, and these two main ingredients are listed here. So the first is the notion of harmonic graph embedment, and I will explain in a couple of words what does it mean. And the second is a notion, is an approach connected to the application of graph grammars, so topological grammars. And this serves for optimize the structure of the graph approximating the data. So what is harmonic embedment? So first of all, as usual, by embedment of the graph into, we mean that each node of the graph is equipped with, is assigned a vector which exists in the data space, and we would like to find such a function phi, which maps nodes of the graph into the data space and dimensional. But we would like to achieve two things. First of all, we would like that this graph would serve as an approximator of the relatively complex data point cloud. And second, we require that this graph would be regular in some sense. And by regularity, we, in this particular approach, we assume harmonicity. So we say that ideal embedment of the graph into the data space must be harmonic. In our approach, harmonic serves as a generalization of smoothness compared to the principal curve approach. But it generalizes this smoothness to arbitrary structure of the graph. So the idea of harmonicity in a way is simple. When we have harmonic embedment, it means that we have such a map phi, which maps each node of the graph in multidimensional space to the mean point of the maps of its neighbors. So it can be applied to any node of the graph besides these terminal nodes. So in this graph, we can introduce a notion of star, and we have two stars, meaning those nodes of the graph which are connected to two neighbors, three stars which are connected to three neighbors, and or we can have four stars, more complex graph, and for each star, we require that the central node of the star would be mapped into the mean point of its neighbors. So the notion of harmonicity is simple. Of course, kind of pure harmonic embedments are sometimes too rigid to require, so we would like to find such an embedment of the graph which would penalize deviation from harmonicity. And to do this, we can also use this spring trick, so analogy with physics. So we can describe for each star of the graph, again, the positive sprints, which will attract nodes of the graph together, and also negative sprints, which will repulse nodes of the graph from each other. So we can continue to reformulate this approach, deviation of harmonicity from harmonicity using this spring language. And in the end, what we do, we formulate a penalty function for the embedment, which we call elastic energy of the graph, and it consists of two parts. So one part describes the stretching of the graph, so it prevents the graph to be actually limits the total length of the edges of the graph in a way, and we have a bending energy of the graph, which describes this deviation from harmonic embedment. So at the same time, of course, we would like that this graph would serve us as a data approximator. So we introduce a mean squared error in a way which is very similar to how you introduce this error in k-means approach. So you say that you would like to minimize the sum of square distances from a data point to the node of the graph, which is the closest to this data point. Okay, so when you sum up all these three things, so the mean square, the errors that you would like to minimize, sum of all edge energies and sum of all star energies, so all these three terms, you have a relatively well-defined optimization problem. And the good news about this optimization problem is that it's piecewise quadratic, very similar to k-means. So you can apply approach which is very similar to k-means. Just unlike k-means, now we have connections between the nodes, which shapes the distribution of the nodes in multidimensional data space. So this was about the first ingredient of our approach. The second ingredient is a topological grammar, use of topological grammars. So the question is how to define, if we know the structure of the graph, we can use this optimization problem to embed it in multidimensional space and achieve good approximation properties while being regular. But how to find the best structure of the graph? This is much more difficult problem. So the idea of topological grammar approach is to start from some simplest graph and define a set of grammar rules, which we can systematically apply to define a set of all possible configurations of graph structures. And so this defines to us a kind of discrete space of possible structures that we would like to test. Of course, I mean, as you know, that the number of such structures, even for simple trees, grows super exponentially. So it's not possible to apply exhaustive enumeration of all the structures and to test elastic energy for all of them. So we have to organize some kind of gradient descent like approach to find the right structure. And this is what we do. So in the end, we have an algorithm of LP, which is called LP graph, elastic principal graphs, which consists of 4 steps. Start from a guess of initial graph. It might be simple, it might be less simple, like in this picture. Then optimize the graph energy and systematically apply all possible graph transformations from a given structure, such that at each point the embedment of the graph is optimized, and we select the embedment which gives the best benefit in terms of elastic energy. So after applying this approach until some stop criterion is met, we will obtain some final graph structure which will be regular in the sense that it will represent a kind of set of connected principal curves approximating the complexity of the dataset. And in the space of structures, what we have is, as I have told you, we have a gradient descent like process of optimization of the structure. So we don't visit all possible structures of the graph. We only visit those which give the best local benefit in terms of elastic energy embedment. So this is how this approach works in some synthetic simulators. So one of these simulators, we call it lizard brain. It represents a branching data distribution in a multidimensional data space in which we know the branching points, so we know the ground truth, so we can estimate how different methods aiming at reproducing this branching complexity find these branching points. And when you look how elastic principal graphs perform with different number of nodes or in different dimensions, you can notice that the main difference between all these methods in the resulting complexity of the graph which is produced. So some methods tend to overbranch and produce many branches which are not really essential, let's say. Or some methods, especially when they are applied in low dimensional spaces, they produce too little branching, and again you miss some important bifurcations in the data. So in this sense, we believe that LP graph represents a nice kind of balance between the complexity of the model as a graph and the complexity of the data. And this is achieved mainly because we have explicit parameters which allow to control complexity of the resulting principal graph approximation. So this ability to control things makes us different from another popular approach for finding graphs approximating the data, namely the approach connecting to the use of minimal spanning tree. And on many examples, we showed that elastic principal graphs are way more kind of regular in terms of number of branches and number of structural elements that they contain. And so, as I told you, in the elastic principal graphs, we have we have methods to control the complexity. But what is complexity of a principal graph? We can talk about geometrical complexity, which is deviation from harmonicity, and we can also talk about structural complexity, which is the number of elements contained in the graph. So these two things are somehow in contradiction to each other. So if you introduce very complex graph in terms of structure, you can achieve perfect harmonicity. But usually we would like to have a balance between these two types of complexity, and to control this balance, we use some tools like this accuracy complexity plots, which represents a return investment trade-off. How complex must be your graph before your geometrical complexity will start to increase too much. And on this graph, sometimes we observe several suboptimal structures, which represent different levels if your branching data distribution is characterized by a hierarchy of branching points. So this is how you can select an optimum, a given level of hierarchy, and kind of evaluate if it satisfies you in your applications. And we also have an explicit parameter in the elastic principal graph, which also controls the structural complexity. So there are many different ways how you can control for appearance of branching points of higher order. We use one particular one, which assigns a penalty to the edges of the graph. And this is a small illustration how this penalty behaves when you start to add branching points of increasing order to the graph. So yet another kind of development of this methodology is dealing with background noise. As usual, those methods which are based on minimal squared error, they are very sensitive to existence of outliers in the data, in particular to background noise. And in the base version of LP graph, you have a possibility of trimming the minimal square error, such that the points which are too distant at the current iteration from any nodes of the graph, they do not contribute to the optimization process. As such, instead of growing a global optimization of data set by a graph, you have a kind of different strategy where you start to grow your graph from, let's say, from local dump of density, and then you expand the graph gradually to the rest of the data distribution, in a sense following the ridges in the density distribution and neglecting the existence of outliers. So the outliers do not have chance to contribute to the final reconstruction of the data. As such, we can deal with a relatively strong background noise, provided that it's close to uniform. Simple illustration of how it behaves, for example, on our synthetic example, where we add more and more noisy points, the kind of the reconstruction of the graph gradually deteriorates, of course, but even when we have 98% of the noisy points, which means that among all these points, only 2% of them, they were initially contained in this synthetic data structure, we can still find the major bifurcations in the ridges of density in this distribution. And this is kind of an extreme when we have 99% of noisy points. Still, we have a hint to the existence of a ridge in the density. And yet another aspect of approximating complex data by complex objects is that we should always control for non-randomness of the structures that we try to approximate in the data, because even in uniformly sampled data sets, we might find some spurious structures which might look like a non-random signal. So in the standard approach to this is use bootstrapping or some sampling And usually what we do, we subsample the data many times, construct the whole ensemble of principal graphs. And in some applications, it's very advantageous because it allows to estimate uncertainty of, let's say, those branching points that we observe and try to quantify from the data. Somehow it becomes a quite common approach in this trajectory analysis in single cell. So in many papers today, we see that indeed, instead of representing one trajectory, we can see the whole ensemble of trajectories, and this already maps the areas where this reconstruction is highly uncertain, and we shouldn't trust, let's say, exact position of the branching points in this area. Just a word that graph-based approximation of the data induce partitioning of the data, and there is one simple trick where we can unglue all branching points of the principal graph. As such, the data starts to be partitioned into partitions which are locally ordered, and so it's a set of local principal curves which makes the approximation. And this notion is relatively, I mean, it's quite different from the standard notion of clusters. So first of all, because you have this local order of data points in each particular partition. And once again, this approach is widely used in analysis of single cell data today. So in the standard tutorials on single cell data analysis, you can detect how this approach is used to define biologically meaningful groups of cells, for example. So, but LP graph is, as I have told you, is a general purpose machine learning method. As you know very well, if you want a machine learning method to shine in a particular application, you have to provide the whole pipeline how this method should prepare the data first and then post-process and visualize in the most meaningful way the results of the application of the machine learning method. And this program is ongoing. It was already done. I mean, and with Luca Pinella, who was the previous presenter, his group developed this stream pipeline in which LP graph serves as a core computational method, but the stream is a completed pipeline for analyzing single cell data. It's user-friendly because it provides a graphical user interface, but it also provides a number of meaningful and insightful data visualizations on top of application of principal graphs. And so currently we collaborate with Luca on extending this and scaling up this approach to really complex multidimensional data distributions connected to the kind of the most recent advances into single cell technology, in particular including multi-omics support, so supporting complex geometry, which is not always can be represented by a simple tree-like topology. Also, we introduce some elements connected to the supervised graph learning when we can use some a priori knowledge to help finding the most optimal structure of the graph. But as I have told you, LP graph is not limited to application to single cell data. Actually, we did apply LP graph to approximate this cosmic web, or we applied it to many other let's say tasks to characterize more or less complex patterns in multidimensional data sets. In particular, this LP graph approach can be applied to bulk tumoral omics measurements, and from our experience, it gives a partitioning of bulk tumoral measurements, which is meaningful from the survival point of view. And but the advantage of this approach is that it defines a kind of pseudo time which might reflect the progression of a given tumor in the process of tumorigenesis. Also, we have a recent application of LP graph to clinical hospital data, which represents a snapshot, synchronic snapshot without follow-up data. So usually when we talk about patient trajectories, we mean that a given patient is followed up for a significant amount of time. But as a matter of fact, in this field of analysis of clinical data, we have much more synchronic data without follow-up. For example, data about hospital stays of patients. And the idea here is that you can also use these concepts of pseudo-time and extract trajectories from the distribution, from multidimensional distribution of patients in this space of clinical characteristics, such that a state of particular patients might be a predictor of the future state of another patient. So if we are able to guess the sequence of states of different patients staying in the same hospital at the same time, for example. So using this approach, we were able to analyze the patient trajectories related to complications after myocardial infarction in cardiology. We also had some results in analysis of diabetes data, and also now we have some applications in COPD field. So, okay, I make a summary here. I started from introducing these density regions. It's a tiny element of describing the complexity of multidimensional data, but it's already extends this simple view on the data, multidimensional data set as a set of clusters. Elastic principal graphs, what it does, it assembles these density filaments into a connected graph. It's based on two principles, harmonicity and application of topological graphs. And there are pipelines which include LP graph as a core algorithm for quantify single cell trajectories. And in the literature today, you can find various generalizations of this idea. So, for example, you can go to the probabilistic field and introduce graph-based mixture models. So this is also exists. So here I would like to acknowledge people who were involved in developing this methodology of, let's say, analyzing data complexity. Of course, Alexander Nikolaevich Gorban at first, and the whole Leicester team. Also my group at Institute Curie, the group of Luca Pinella, collaboration with him was a fantastic experience. At this stage, I would like to invite you to ask questions if you have any.
Thank you very much, Andrei. So whilst I'm waiting for a rising hand, I would like to ask you a question, or maybe a couple, if I may. So in this framework which you have, like you basically approximate data with the graph. So I was wondering that maybe for some applications, the information about the density along branches could be important, but I haven't seen in your examples so that this information somehow propagates into the structure of the graph. Could you please comment on that?
So if I understood your question right, is that let's say the fluctuations in density of multidimensional data distribution must be, in your point of view, reflected in the structure of the graph?
Well, I'm not saying must be, but sometimes it may be useful.
Sometimes it might be useful, yes. Let us say we can compare these graph-based approximations with so-called self-organizing maps. And I think what people did when designing the self-organizing maps and when they apply them, that they have exactly this idea in mind, that more dense regions of the data space must be covered by more nodes at least, or more tightly. In LP graphs, the idea is a bit different. The idea is indeed kind of introduce more uniform, let's say, geodesic distance, which would be maybe less sensitive to the fluctuations in data density. But of course, fluctuations in data density, they do affect the structure of the graph. So in particular, they start to branch more extensively in dense regions.
Yeah, because like when I was looking at your sort of stream and other sort of trajectories, for example, I was thinking that since you're somewhat alluding to that, there is somehow the ordering, or maybe even, let's say, a temporal structure, right? In the data, like it would be nice to have a tool that would enable us to tick more often in certain regions, where the time flies faster, right?
Yes.
Or like maybe slower in some other regions. So that was my sort of curiosity, basically, and then that's the origin of my question.
It's an interesting question, which might be related indeed to the relation between pseudo time and physical time. That indeed, if the speed of the process is highly unequal in different parts of the space, indeed, if it's close to kind of steady state, it might kind of produce we might observe objects more frequently in these regions of the space, which might lead to indeed increase in density. So density might be used to indeed to find this relation between pseudo time and the physical time. I do agree with you.
Yeah, and then the second question was that like these examples which you have, they have very clear somehow maybe lower dimensional, as you said, like ridges in them. I'm curious, like what the methods would do if there are no ridges of the type. Like what's going to happen?
Well, the simple answer is, I mean, maybe you should use other approximators where the data set is not characterized by existence of these filaments or ridges. Maybe it's just not a good approximator in this case. So if, I mean, what we observe, if our data set is locally very, very strongly deviates from this assumption of, let's say, local one-dimensional structure, intrinsically one-dimensional structure, of course, it produces many unstable branchings. And in single-cell field, it's very difficult to interpret them. So it might. So I think what is important is to detect such situations and maybe weight them less when at the interpretation step, for example. This is what we do in applications.
Yeah, and like if I may, there is a third question, which is absolutely like wild now. Like you know that in approximation theory, when there is this famous bound by Roy DeVore, I believe, who first somehow broke any hopes of escaping the curse of dimensionality by saying, look, like in some like very regular situations, you just cannot break through the curse of dimensionality, given that, let's say, if the domain of your function is, say, multidimensional, then unfortunately there will have to be exponentially many elements in your approximation in terms of the worst case scenario. So seeing now that, and then the criticism for that was, for example, this, why don't I use, let's say, a space filling curve to substitute my problem with the, say, one-dimensional problem? And of course, that gives rise this sort of tricks when I refuse to work with high dimension and do the space filling curve business. Of course, I become quite unstable, right? Because I'm exploring all that region and like, and then of course things may happen. Now, the question to you: do you envision that this stuff may actually be bridged to close the gap? And I think Alexander Nikolaevich wants to comment, I think, or maybe completely discredit my question. So, so go ahead.
But just my quick feedback on this. There's a simple question. I don't know. The kind of a bit more elaborated question is that we had this idea in mind. I mean, similar idea in mind when we introduced this lizard brain term, with having in mind that indeed globally the data set might be high dimensional, still locally it might be characterized by very small dimensions. So there might be some kind of this situation when you have a big discrepancy between global dimensionality and the local dimensionality. So this is what you seem to refer to.
Yes, and I think in your case you basically found a very regular tool to achieve that aim, right?
Yeah,
so
the LP graph is indeed, in this case, LP graph can be a part of toolbox for dealing with such distributions. Indeed.
Yeah.
Okay.
Alexander Nikolaevich, I think you are burning to comment.
Yes, I'm just so that is this method. Of course, you told completely correctly that this is high dimensional, low dimensional interplay. So there are two methods to work with two classical, the ancient methods to work with high dimensional and low data. First of all, has 120 plus years in principal component analysis, and the second has like 70 or 80 years is k-means. And these are just two ways of data approximation. The main idea is data approximation, but because we have so bad situation with high dimension, we need something more or less rigid. And principal components are too rich, just beams. And k-means are too flexible. They lose old structures, just small finite sets. And there's interplay between small finite sets and straight beams. Well, there are some ideas of bending. This is principal manifold. But topologically is not the best idea. Idea of space filling curve works for some problems, but not for that, because it absolutely loses all the geometry. There's no smoothness, no geometry, just space filling. It's not. Of course, this sort of coding may be useful, but not in general, because it loses geometry. And this graph is an interplay between absolutely flexible k-means, finite, structureless finite sets, and absolutely rigid principal components. And the key idea is harmonicity. What instead of straight line, the good regular graphs with regular stars inside. What does mean regular? Means that average of vertices of a star is the middle. Of course, this sort of instead a straight line consists of two stars, of course, because it's linear. And so this is, and that is, well, there could be some, and then, of course, deviations from this ideal picture should be penalized. Deviation from the straight line should be penalized. Deviation from the harmonicity should be penalized. Too many nodes, deviations for small finite sets should be penalized. This is the simple, absolutely simple set of ideas. And of course, this, and then is one more trick, because algorithmic complexity could be huge even from not very large graphs. Because complexity of a graph grows exponentially with the number of vertices. Therefore, we should create the achievable, algorithmically achievable graphs, what we can computationally not too complex. And therefore, this computational complexity is also penalized in the language of topological grammars. That's all. That's fine. So this is, I think that, okay, thank you. I just comment because I have
Thank you very much. Thank you very much.
Have been mentioned as a somehow how a co-author of this approach. Okay, thank you.
Thank you.
Okay, so I guess we can stop at this point. I would like to thank all speakers once again, maybe upload once again.
Thank you.
Thank you.
I hope you enjoyed and learned something during this workshop. And so have a nice rest of the day. Bye bye.
Thank you, Andrei and Ivan, for organization. Thank you, Luca and Kees, for talks. And this is Ivan and Andrei for talks. Thank you, guys.
Thank you very much. It offered so much food for thought today, as never before, almost. Thank you.
Yeah,
Thank you.
Thank you.
I think this summer we will organize a 2-day workshop about learning in high dimension with several big masters involved in additional classes. Of course, we are the biggest master, but somebody else will be also involved. Okay, next summer.
Thank you.
Thank you.
Bye now.
Bye.
Bye bye.
Bye bye. Спасибо, Андрей. Спасибо.
Да, с удовольствием.
Давай. Ну, пока. Все, я выхожу.
Ага. До свидания. Ага. Свяжемся.